Bom dia, todo mundo. Bem-vindo ao segundo dia do GBMeeting. Espero que todos estejam me ouvindo. Qualquer problema aí, só acionar, se não estiver me ouvindo direito. Bem, hoje eu gostaria de iniciar com alguns recados antes de dar início às palestras. Principalmente aos avaliadores. Ontem a gente teve um problema técnico na plataforma. A plataforma começou a apresentar grande estabilidade durante a tarde, conforme foi enviando, os trabalhos foram sendo avaliados. A gente conseguiu arrumar, mas infelizmente o que houve foi que a gente perdeu as avaliações que já haviam sido feitas. Então a gente pede desculpas em nome da comissão, estou aqui pedindo desculpas pelo esse pequeno erro técnico e pedir encarecidamente que os avaliadores refaçam, então, no sistema da Even, a avaliação foi feita ontem. O problema foi resolvido, menos mal, ainda bem que foi no primeiro dia de evento, ainda tem 2 dias aí de avaliação, mas felizmente a gente conseguiu resolver esse problema, peço desculpas, tá bom? Lembrando que a gente sempre está respondendo o mais breve possível por e-mail, então qualquer dificuldade, só entrar em contato. Seguindo agora com o tema de hoje, a gente vai começar então com a mesa redonda, né? O tema vai ser divulgação científica. Começando então com as damas, eu gostaria de convidar a doutora Natácia.
Tudo bem, olá Natácia? A todos. Oi, Ciro. Bom dia, gente.
Bom dia. Fazendo então uma pequena apresentação, ela é criadora do Entenda Genética, é formada em Biologia e mestre em Genética e Melhoramento de Plantas pela UFG, possui doutorado pela Faculdade de Medicina de Ribeirão Preto, na USP, com um período sanduíche no Departamento de Agricultura dos Estados Unidos, USDA. Seu projeto de divulgação científica, o Entenda Genética, tem como objetivo discutir sobre análise e interpretação de dados genéticos. Bem-vinda, Natácia, mais uma vez.
Muito obrigada.
Imagina. Seguindo, então, aqui com as apresentações, gostaria de chamar o professor Eduardo. Está aqui agora na tela, tá certinho. Você está no mudo. Um minuto que eu já... Oxe, no caso aqui, o microfone não está funcionando. Deixa eu entender por quê. Eduardo. Você ainda está no mudo. Bem, enquanto a gente tenta resolver isso, já vou chamar também o próximo convidado. Vou colocar aqui o Frederico, professor Dr. Frederico, bem-vindo. Continuando com as apresentações, ele é editor do blog Omixdata, que tem por objetivo divulgar conteúdos de biotecnologia, bioinformática e aprendizado de máquina. Bacharel, mestre e doutor em biotecnologia pela Universidade Federal de Pelotas, graduado em análise e desenvolvimento de sistemas pela Faculdade de Tecnologia Senac, no Rio Grande do Sul. Atua como professor e pesquisador nessa mesma universidade. Possui experiência com bioinformática aplicada e análise de dados de NGS, desenvolvimento de aplicações com Python, Data Science e Machine Learning. Bem-vindo, Dr. Frederico.
Muito obrigado.
E seguindo agora para o último, mas não menos importante, Eduardo Castan. Ele é criador do Universo da Biologia Molecular, mestre e doutor em Genética e Melhoramento Animal pela Unesp, com projetos desenvolvidos na Universidade de Melbourne, Austrália, e na Universidade de Harvard, nos Estados Unidos. Possui MBA em Gestão Empresarial pela FGV e atuou por 7 anos na Thermo Fisher Scientific. Possui 15 anos de experiência com técnicas de biologia molecular, trabalhando como especialista em PCR em tempo real e sequenciamento de nova geração. Por meio do Universo da Biologia Molecular, atua como professor divulgador científico, utilizando as principais redes sociais. Bem-vindo, Eduardo, também ao nosso evento. Em nome da
Valeu,
comissão,
Ciro.
Agradeço.
Obrigado,
Imagina.
Cara. Bom dia, pessoal.
Então, como que vai ser a dinâmica aqui dessa mesa redonda? Eu vou passar a palavra para vocês se apresentarem também, além dessa breve introdução, se vocês quiserem falar um pouquinho, abrir o tema, então fiquem à vontade. Esses primeiros minutos é mais para se apresentar, dar um espaço, e depois a ideia é que, conforme forem surgindo perguntas da plateia, a gente vai jogando então aqui na tela e a gente vai conversando sobre o tema de divulgação científica, tá bom? Vou fazer agora então ordem alfabética. Então, Eduardo, fica à vontade, a palavra é sua.
Beleza, gente. Bom dia. Vou tirar o fone aqui para conseguir me escutar um pouco. Tudo bem? Como é que estamos aí? Cara, Ciro, eu não sei exatamente o que vocês precisam saber, mas acho que mais do que falar das coisas que eu fiz ano passado, acho que mais legal hoje mesmo é falar do universo da biologia molecular, né? No universo da biomol, que surgiu um pouco mais de 2 anos, não sei se quem está aqui já ouviu falar, conhece o Universo da Biologia Molecular, mas o Universo da Biologia Molecular é um ambiente, uma comunidade para troca de conhecimento sobre biologia molecular, temas relacionados, que basicamente está no YouTube, no canal do Universo da Biologia Molecular, que tem aulas toda segunda-feira sobre esse tema. De vez em quando, algum tema um pouco fora disso. Por exemplo, teve aí, sei lá, acho que alguns dias atrás aí, que eu falei do teste genético para sexagem de embrião. Então, assim, não é sobre a técnica de biologia molecular diretamente, mas está relacionado a um. E é isso. Então, é um ambiente ali para aulas. Para quem quiser aprender, pode entrar lá gratuitamente para aprender. E tem o canal, e tem o perfil também no Instagram, que surgiu inicialmente para dar apoio ao canal do YouTube, mas ele criou vida própria, porque o ambiente lá é diferente, e ali hoje tem conteúdos específicos e exclusivos que estão ali também. Mas no final das contas é isso, é uma divulgação científica / educação, só que para um público um pouco diferente, né? O meu conteúdo, pelo menos nessas redes, terão outras, depois eu falo sobre isso, mas nessas redes, nesses canais de comunicação, eles são focados para as pessoas que querem aprender biologia molecular. Então, geralmente, é alguém que pelo menos está na graduação. Tem gente que nem entrou na graduação ainda, tem, mas a maioria está na graduação, na pós-graduação, ou já é formado. Então, é um nível de divulgação científica um pouco diferente do que falar com o público em geral, né?
Bem legal. Acho que vai ser interessante essa conversa, que a gente vai ver também como diferente de divulgação científica para quem, né? Então, acho que nessa conversa depois, conforme a gente for seguindo, vai evoluir bastante. Frederico, a palavra é contigo, fica à vontade.
Bom, da mesma forma que com o Eduardo, né, eu também criei o Omix originalmente para servir como uma divulgação científica já para um público que já tem um certo nível de conhecimento assim, técnico sobre alguns conceitos. Então, dificilmente alguém que não está num contexto de um curso de graduação, pelo menos assim, em biologia ou em computação ou alguma área assim afim, vai buscar conteúdo sobre bioinformática, né, que é uma coisa bem mais específica, né, mais nichada. Então, o Omix, ele meio que surgiu assim com a ideia de ser uma forma de se disponibilizar tutoriais, disponibilizar conteúdos de tópicos que são difíceis de se... Pelo menos assim, até um tempo atrás era mais difícil de se encontrar em português ou alguma coisa, né? Montagem de tutoriais mesmo de montagem de genomas, né? Até tem alguns textos, né, que falam de montagem de genomas mais conceitualmente, ou anotação de genomas mais conceitualmente, mas não tinha nenhum tutorial ou alguma coisa que te ensinava como fazer de fato, né? Como rodar um programa, como às vezes. Então essa foi a minha principal motivação, assim. Logo no começo do projeto eu pensei quantas vezes eu quebrei a cabeça vendo fóruns ou lendo documentações obscuras ou, né, e tentando às vezes ler o código do próprio programa, tipo o código-fonte, para entender por que ele não estava funcionando. E a ideia do Omix era que tivesse um local onde as pessoas pudessem ter acesso a esses tutoriais, né, de uma forma mais amigável, um pouco mais explicada, assim, principalmente para quem está começando na área. Mas isso é um público nichado, né? Então, a gente tem... Claro que tem outros posts, né, que não são tão técnicos, assim, que eles também acabam sendo mais acessíveis para quem é de fora, né? Para quem que às vezes tem curiosidade na área, já ouviu falar, mas quer entender algum conceito, né? Então, tópicos bastante em alta, né? Tipo, agora a gente teve o AlphaFold, mas da mesma forma ainda assim é uma divulgação assim para o público com um certo nível de conhecimento já, de familiaridade, né?
Sim. Muito obrigado, Frederico. Natácia, sua vez, falar um pouquinho então aí do seu papel divulgadora científica, fica à vontade.
Olá, gente, bom dia a todos. Olá, olá. Posso compartilhar uma apresentação, então, 5 minutinhos?
Claro, pode, esse minuto inicial, rapidinho, não tem problema não.
Então esse primeiro momento é a apresentação do que eu faço, certo?
De cada... Isso,
então,
exatamente.
Beleza. Então,
para depois entrar.
Imagina. Beleza. Gente, eu sou uma pessoa muito visual. Aqui. Beleza, então vamos lá. 2 segundinhos.
Imagina, sem problema. Coloca aqui na tela.
Tá dando para ver a apresentação? Que daí eu já vou maximizar.
Ainda
aqui.
não, ela demora um pouquinho mesmo. Daqui
Beleza.
a pouco aparece para mim e a gente adiciona. Não se preocupe.
Tá bom.
Ó, já teve gente elogiando aqui. Vou até colocar aqui na tela, já tem gente comentando aqui. Ó. Bom dia, aprendi muito com os vídeos do Universo da Biologia Molecular.
Ô, Bárbara, que bom. Obrigado pela mensagem. Fico feliz demais.
Natácia, ainda não está compartilhando. Ah, tá, agora entrou. Calma aí. Beleza. Eu vou colocar agora no principal. E agora tá, mas agora é só colocar em modo apresentação pra gente.
Beleza, ótimo. Então.
Foi, tá perfeito. Fica à vontade.
Gente, muito obrigada, então, pelo convite de vocês para falar sobre divulgação científica, que é um tema, assim, que eu absolutamente sou apaixonada. E eu quero conversar com vocês, primeiro, o que eu entendo que é a divulgação científica para a gente estar na mesma página. Então, para mim, é um compartilhamento da ciência. Então, é uma série de iniciativas que nós temos que a gente pode, então, tornar o pensamento e o conhecimento científico acessíveis. Aqui a gente viu que o Fred e que o Eduardo, eles têm diferentes perspectivas, né? Às vezes a gente pensa na divulgação científica só para o público, digamos assim, entre aspas, leigo, mas não necessariamente. Então, eu vou conversar com vocês sobre algumas experiências que eu tenho dentro da divulgação científica, porque às vezes você vai se sentir mais confortável em um âmbito do que em outro, e assim a gente pode discutir melhor, tá bom? Então, a primeira coisa que eu quero dizer para você, quando a gente está trabalhando com a divulgação científica, é que você conheça o universo da persona que você está trabalhando. Cada persona tem um universo distinto. Então, imagine a persona como algo fictício, sabe? Uma personalidade fictícia. Eu quero falar com criança, eu quero falar com adulto, eu quero falar com público especializado. Você tem que conhecer esse universo. E aí eu vou trazer para vocês alguns casos, bem rapidinho, não vou aprofundar em nada, que eu tive aqui para compartilhar com vocês dentro da divulgação científica. O primeiro caso que eu vou compartilhar é o caso do coração. E aí você deve estar se perguntando, como assim o caso do coração? Eu sou apaixonada por criança, mas hoje eu atuo dentro da genética, com o meu público também especializado, mas eu também tenho outras frentes de trabalho. Isso aqui, obviamente, antes da pandemia, é uma fila de menino doido querendo pintar o rosto. E por que eles estavam assim, doidos querendo pintar um rosto, né? Porque, gente, eles descobriram que o coração de verdade não era no formato coração que contaram para eles, e eles ficaram muito revoltados com isso, porque a maioria não sabia que o coração não tinha o formato que eles viam na televisão, que os pais desenhavam. Eles ficaram tão revoltados, e assim foi a forma que eu entrei dentro do universo deles para explicar como é que funcionava o coração, e dentro desse contexto, eu consegui também entrar dentro da genética, mas a gente não vai entrar aqui, porque a gente vai puxando, a gente vai entrando no contexto. Aqui eu entrei num contexto lúdico. Então, as crianças, elas saíam com a mãozinha, olha aqui é o coração de verdade ou de mentira. E teve uma criança que ficou muito revoltada, porque os pais mentiram para ela. Então, assim a gente conseguiu entrar dentro desse universo. Depois eu tenho o caso do turismo para contar para vocês, que foi um trabalho que a gente desenvolveu dentro de Brasília, considerando um turismo científico. Então, olha só, aqui já mudou completamente a persona. Brasília é tida como uma cidade educadora. Então, o que foi feito dentro deste projeto? Nós pegamos os locais de divulgação científica que tem na cidade, e aí a gente foi mapear esses locais e nós fizemos um aplicativo. Então, a pessoa podia pegar um aplicativo e identificar qual local ela gostaria de visitar e, assim, estar inclusa dentro dessa cidade educadora. Além disso, nós fizemos um trabalho para identificar o que acontecia dentro desses espaços de divulgação científica. Então, isso foi feito a partir de uma abordagem em que eu identificava as oportunidades, as fraquezas, as forças, as ameaças que os coordenadores desses espaços observavam e que os trabalhadores observavam, porque é diferente, né? Às vezes a gente está trabalhando com uma coisa na gestão e tem desafios que nós não enxergamos dentro de quem está ali no dia a dia colocando a mão na massa. Então, esse foi um outro universo, um outro nicho. Depois disso, eu tenho um outro caso, que é o caso da inclusão, porque quando a gente fala de divulgação científica, às vezes a gente esquece que é importante também incluir dentro da sala de aula. Lembra que eu falei para você que é uma conexão, é uma forma de divulgar em diferentes ambientes? Então, aqui eu desenvolvi, dentro da disciplina de genética de populações, uma metodologia para a prática, tá? Aqui o que a gente estava querendo fazer era um cenáriozinho de evolução, considerando tamanhos de bicos, de passarinho e diferentes sementes. A gente pode conversar sobre esse trabalho depois, mas por que ele é um trabalho inclusivo? Porque ele foi o primeiro trabalho didático em que nós não incluímos apenas a habilidade de mobilidade das mãos. Por quê? Porque eu tinha uma aluna que ela não conseguia mexer com as mãos. Então, a gente fez biquinhos de papel, mas nós também fizemos biquinhos para pegar aquelas sementinhas com imã. Então, essa aluna minha, que não conseguia mexer, ela sempre estava excluída das atividades, né? E a gente traz essa forma de divulgar, trazendo o aluno para dentro do contexto. Por fim, aqui a minha experiência, né, no caso das redes sociais, que por um acaso eu acabei entrando, e eu estimulo todo mundo a ter essa experiência, gente, porque nós somos comunicadores. Quando nós trabalhamos com pesquisa e ciência, nós somos comunicadores natos, e aí saiu, então, o Entenda Genética. E eu estou trazendo esse último caso aqui, que é a minha experiência mais atual com a divulgação científica, para dizer para você que, dentro do Entenda Genética, eu também tenho um público que é mais especializado, então perceba, já é outro tipo de persona, e nesse ambiente o meu objetivo é ensinar as pessoas a pensarem em suas perguntas científicas, a analisarem os seus dados genéticos, interpretarem os dados genéticos, e a gente também faz consultorias gratuitas disponíveis no YouTube. Então a gente está tanto no YouTube, Facebook, quanto no Instagram, e é muito interessante, porque aqui, nesse caso, a minha persona é o público de graduação, pós-graduação, professor, técnico, e quem eu chamo de aprendedores, que a gente nem imagina, mas que estão inclusos dentro desse ambiente também. Então, a gente consegue, a partir disso aqui, expandir para vários outros locais, para vários outros países que a gente nem imaginaria. E ontem eu estava olhando uma discussão no chat, eu achei bem interessante, né, aquela questão, mas será que a gente tem que trazer o pessoal para dentro da rede social, ou a gente tem que expandir? Eu acho que cada caso é um caso, cada persona é uma persona, e é bem gratificante quando a gente também consegue expandir o nosso conhecimento para fora dos muros, né? E eu trouxe esse exemplo aqui, eu acho super legal, de um senhor que resolveu fazer biologia em cima do canal de genética. Então, eu falei para você que era para aprendedores, né? Porque também não necessariamente você vai captar só o público tão especializado assim. Outra coisa interessante é que a gente acaba ajudando pessoas que não necessariamente, apesar de estarem na academia, elas têm um acesso à informação. Então, por exemplo, eu tenho um caso de uma menina que tinha comprado um monte de primer para RAPD, amplificação de RAPD, mas ela queria amplificar microsatélites e não dava certo. Então, pensa bem, é uma questão conceitual que foi resolvida ali. E também a gente encontra algumas figuras, né? Eu trouxe aqui para vocês, estou compartilhando, um que eu achei bem curioso, né? Então, o rapaz me perguntou se eu achava que era possível mudar a estrutura do DNA dele colocando uma aranha que já tem no DNA, e que ele levava isso muito a sério. E, gente, ele colocou isso em um monte de vídeo meu. Eu falei, eu acho que ele não tá zoando com a minha cara, eu acho que ele tá falando sério mesmo. Aí eu fui lá, respondi para ele, e eu realmente acho que ele estava precisando da resposta, né? Porque ele agradeceu bastante. Então, não negligenciar esse tipo de dúvida que chega até a gente, porque de repente a gente vai com aquele olhar crítico, né? Mas a pessoa está cheia de fome de conhecimento. Dentro do Instagram, assim como o Eduardo falou, o meu público é um pouquinho diferente, então a população também vem até mim procurando sobre questões de testes genéticos e outras coisas. Tem muitas histórias bacanas dentro deste contexto. Mas, independente da persona, do universo que você vai entrar, eu quero dizer para você que você escute. Quando a gente escuta o outro, nós conseguimos, então, atender às necessidades da sociedade, que é o principal papel da divulgação científica. Lembrando que nós temos uma sociedade heterogênea, então você pode entrar em diversas abordagens com encanto. Então, sempre tem isso na sua mente. Quando você for trabalhar com a divulgação científica, leve isso com encanto, escute o que as pessoas estão falando, mas principalmente conheça o seu universo. No começo da nossa conversa, eu falei para você conhecer o universo do outro, mas você também precisa conhecer o seu universo para exercer a empatia, para conseguir se comunicar e comunicar dentro daquilo que você se sente confortável, porque também tem que ser prazeroso. Por fim, termino aqui minhas palavras com uma frase do Ariano Suassuna que eu gosto muito: como sou pouco e sei pouco, faço o pouco que me cabe me dando por inteiro. E é isso, gente. Muito obrigada pela oportunidade de compartilhar com vocês um pouquinho do meu trabalho. Eu não poderia deixar de trazer para vocês essa experiência que eu tive, porque eu acho que, de repente, pode conversar aí com outras pessoas que acredito que tenham também outras fomes de abordagem, né? Não necessariamente para um público específico, ou para um público infantil, ou para um público adulto, e assim por diante. Muito obrigada.
Obrigado você, Natácia. Eu acho que assim como 3 divulgadores científicos que a gente tem hoje aí, como obrigação, eu peço para toda a plateia já começar pelo menos seguir, curtir, se inscrever no canal, curtir esse vídeo também. Então, depois eu vou até pedir aí para todos os divulgadores, passem depois de novo o nome, a gente põe nos comentários para todo mundo seguir vocês nas redes sociais. Acho que é o mínimo como cientista, a gente dá apoio aí para pessoas que fazem divulgação científica. A ideia agora é continuar, eu vou mostrando alguns comentários, então a gente tem até algumas perguntas que já começaram a aparecer aqui, e aí a gente vai conversando sobre, a ideia é, um vai complementando, não precisa necessariamente todo mundo responder, se alguém quiser complementar, vai falando, a ideia é realmente tornar isso aqui bem dinâmico, uma conversa com a participação aí de todos, tá bom? Vou colocar a primeira pergunta, que já são 2 em 1, tá? Então a pergunta é o seguinte, do Ricardo Zé Pimenta. Vocês perceberam uma maior procura pelos conteúdos de vocês após o início da pandemia? Essa é a primeira parte. A segunda é como vocês já têm esse público mais especializado, houve alguma adaptação para entender um público mais leigo? Então, o que vocês acham dessa questão da procura e se depois vocês tiverem que fazer alguma adaptação por aumento da procura, alguma coisa nesse sentido? Frederico, se você quiser começar, aí a gente vai conversando e vai complementando conforme os assuntos vão surgindo aí, tá bom?
Certo. A gente recebeu algumas procuras, assim, mais relacionadas à parte de bioinformática, assim, então, análise de dados de sequenciamento, né? Agora, com o caso do coronavírus, se ficou bem popular esse sequenciamento com nanopore, né? Porque, enfim, a tecnologia ela está mais madura e ela foi usada em larga escala agora para os diferentes isolados, né? Então a gente fez tutoriais mostrando como se faz essa análise, mostrando que não tem muita diferença assim, em relação às tecnologias de segunda geração de sequenciamento. E essa foi a principal, assim, talvez o principal ponto assim, que a gente abordou com relação ao coronavírus. A gente não teve muita demanda para materiais de divulgação sobre a doença em si, né, e sobre todos os cuidados, até porque isso é algo que já estava sendo bastante ventilado por outros divulgadores que até têm mais propriedade para falar sobre isso. Como o nosso foco é mais na área de bioinformática e genômica mesmo, fazia sentido para a gente se ater a essa parte assim, do tema.
Certeza. Assim. Sim. Eduardo, Natácia, se quiserem complementar a pergunta, fiquem à vontade.
Ô, Ricardo, sua pergunta é boa, cara. As duas partes são boas. Mas eu tenho dificuldade para responder a primeira, né? Se teve mais procura no começo da pandemia. É difícil para mim falar, é difícil para explicar isso, porque o Universo da Biologia começou com a pandemia. Ele começou junto com... o Universo da Biologia, o primeiro vídeo foi ao ar em dezembro de 2019, então foi, cara, quase junto, né? O que eu sinto, o que eu consigo te falar é assim, agora que as coisas começaram a abrir, que as pessoas estão vacinadas, está todo mundo saco cheio do digital, né? Teve aquela ressaca do digital, eu senti uma certa resistência aos eventos ao vivo, online, que eu faço, eu senti um pouco mais de dificuldade. Mas também não é fácil de falar, porque o Instagram, que é a minha maior fonte de divulgação, né, onde eu pago anúncio para convidar as pessoas para participarem, ficou caro. Com o aumento do dólar, com a procura pelo digital, começou a ficar mais caro fazer esse tipo de anúncio. Então tem um pouco da desaceleração aí nesse ponto. Mas eu acho que sim. Eu acho que, entre aspas, eu dei um pouco de sorte de começar o Universo da Biomol na pandemia e favoreceu, né? Eu estar online, as pessoas estarem online também. Agora, a outra parte da pergunta, né? Aí você falou assim, né? Como vocês já têm esse público mais especializado, houve alguma adaptação para entender um público mais leigo? Houve, Ricardo. Acho que essa parte da pergunta é a melhor. Porque eu comecei a falar sobre biomol, que é bem nichado, né? É bem específico, apesar de ser amplo para qualquer pessoa que trabalha na ciência da vida, mas tem que trabalhar nessa área. Então eu não pego assim, sua tia-avó, por exemplo, que não trabalha com isso. E eu comecei a fazer uns testes de colocar conteúdos leigos. Então tem esse aí do caso do bebê, eu coloquei do caso dos humanos geneticamente modificados, eu coloquei alguns posts no Instagram também, eu fui jogando. E todos eles, ou a maior parte deles, teve um desempenho muito abaixo, muito menor do que o conteúdo técnico de biologia molecular. E eu fiquei brigando com isso Eu fiquei brigando com isso por uns 6 meses. Falei, não é possível, esse
Olha
tema
só.
aqui é interessante pra caramba. Quem não quer saber de bebês que foram geneticamente modificados lá na China? Puta negócio legal, né? Mas ainda assim o desempenho era bem menor. E aí eu decidi fazer uma coisa, separar os canais. Então eu tenho o Universo da Biomol agora, né, que eu já apresentei, e um que vai ser lançado agora, eu nem falei sobre ele, nem divulguei, vocês estão os primeiros a saber. A única coisa que eu coloquei foi um trailerzinho ontem,
Colocar uma
um
musiquinha
spoiler.
inédita aqui.
Isso, que é o Nem Tudo na Vida Genética. É um canal de divulgação científica puro mesmo, para público leigo, para falar com as pessoas, com a sua tia-avó, por exemplo, que não sabe o que é PCE, que não sabe o que é nada, entendeu? Aí teve essa necessidade. Isso é a importância de você falar, como a Natácia começou, conversar com o seu público-alvo mesmo, com a sua persona. Senão você está falando com ninguém, você está falando com quem não quer te ouvir.
Perfeito, obrigado, Eduardo. Natácia?
Gente, concordo com o Frederico e com o Eduardo. Eu enfrento o mesmo problema, assim, porque o nosso público é muito específico, então não adianta colocar alguma coisa ali que não vai ter tração. Eu estou ensaiando abrir um outro canal um pouco mais suave para eu conversar com um público que tem o interesse, mas que não é especializado. Confesso que estou ensaiando. Admiro o Eduardo, admiro, porque dá um trabalho do caramba fazer o negócio. Mas, assim, o que eu fiz, né, dentro do Entenda Genética? O Entenda Genética ele também nasceu na pandemia. São canais filhos da pandemia, né? O primeiro vídeo foi em janeiro. Eu não imaginei que fosse crescer. Inclusive, o primeiro vídeo, eu acho que eu estava na fazenda, porque eu sou de Goiás. E falei, o pessoal pergunta tanto o que é filogeografia, deixa eu fazer um negócio aqui. Foi gravado debaixo de árvore. E o negócio foi crescendo. Depois, realmente surgiu essa necessidade, o pessoal perguntando muito, né, para tentar entender aquela linguagem, até mesmo dentro dos acadêmicos. Aí eu fiz um vídeo explicando sobre a doença e tudo mais, sobre o vírus, um pouco especializado, mas mais para aquelas pessoas que não entendiam muito bem os conceitos, bem suave, e coloquei no meu canal e também dei palestras sobre isso em outras universidades, e assim eu fiz dentro do contexto da pandemia, com a busca que me chegou, mas eu não produzi esse tipo de material, porque o meu público, assim como o do Eduardo e do Frederico, é bem específico, tá bom?
Eu acho que é um ponto, né? Porque eu lembro quando a gente fez... A divulgação científica é sempre um tema que a gente traz aqui no evento, né? Então, nesse evento de encontro de pós-graduação, a gente sempre gosta de trazer divulgação científica e também exaltar profissionais da área que têm esse papel árduo e tão glorioso, eu acho mesmo, de você pegar a divulgação científica e conseguir traduzir isso para outras pessoas, seja um público mais especializado ou não. E aí, ano passado, a gente trouxe pessoas que eram canais mais para um público mais leigo. Aí a gente estava, esse ano, falou assim, não, vamos mudar um pouquinho, chacoalhar as coisas, né? Sempre tem que dar uma chacoalhada, vamos tentar trazer um público aí, canais que falem com um público mais especializado, e assim a gente consegue também ver um outro cenário aí da divulgação científica. Então, acho que está sendo bem interessante você ver também que é possível você fazer divulgação científica além das pessoas para leigo. Você pode voltar o seu público para um público mais especializado. Então, acho bem interessante. Eu vou colocar a próxima pergunta agora, que é do William. Doutora Natácia, quais aspectos você acha mais importantes na adaptação do discurso entre o público leigo e o mais envolvido com a ciência? Então, já puxando essa pergunta aí que eu estava agora, acho que esse comentário que eu fiz, essa pergunta cabe bem. O que você tem a dizer, Natácia? Fica à vontade.
Gente, eu sempre gosto de falar que a gente tem que entrar no universo da pessoa. Para fazer essa adaptação, você tem que entrar no universo. Por isso que eu falei assim, que a divulgação científica ela também é um exercício de empatia, né? Frederico mesmo disse, eu tive dificuldade, então eu resolvi criar, porque foi muito difícil para mim. Então, assim, o que eu faria se eu fosse adaptar os discursos, tá? Minha sugestão. Você vai pegar as principais dores dessas pessoas que você quer chegar. Porque essas dores, elas são diferentes, tá? Então, cada um anseia algo. Então, quais são as principais dores que elas têm? O que elas querem saber? A partir disso, você consegue adaptar o discurso. Então, eu sei que parece estranho, mas lá naquele exemplo do coração, eu consegui chegar na genética, tá? Por quê? Porque eu acessei o mundo lúdico daquela criança utilizando a pintura. Então, é a forma que eu consigo conversar com elas. Então, eu acredito que é isso. Você vai pegar as principais dores, as principais aspirações que esse público tem, e a partir disso, você vai começar a entrar no universo que você quer conversar e adaptar o seu discurso a cada uma dessas pessoas, tá bom?
Perfeito. Aproveitando também, né, Eduardo, Frederico, essa mesma pergunta, vocês acham que tem uma diferença? Por exemplo, eu estou até aqui matutando essa questão? Há uma preocupação maior por você estar falando com um público mais especializado? Vocês acham que vocês se cobram mais com medo ali de estar falando alguma coisa errada, que você sabe que está falando com alguém que já é um público diferente? Vocês acham que essa adaptação exige mais para ir por mais estudo do que se você fosse falar com leigos? Então, complementando essa pergunta, o que vocês sentem aí de diferença? Ainda mais o Eduardo falou que agora vai ter 2 canais entre esses 2 discursos aí. Fiquem à vontade aí para complementar a resposta da Natácia.
Bom, Fred, se quiser começar, fica à vontade aí. Mas
não, pode.
Tá, eu tenho bastante preocupação com o meu conteúdo, porque o meu pensamento... Eu não diria assim que quando eu fizer o conteúdo para pessoas leigas, já fiz alguns, né? Mas quando o canal estiver mais pronto para isso, eu vou dar menos importância ao nível do conteúdo. Não vou, vai ser o mesmo nível de informação com bastante cuidado para produzir. Mas o caso do universo da biologia molecular, como eu estou falando com pessoas que muitas vezes já entendem do assunto e vão utilizar aquilo na vida, no dia a dia delas, né? Não é simplesmente estar ali absorvendo uma informação, tipo assistindo um jornal nacional e... Não, aquilo que eu estou ensinando ali, ela muitas vezes vai meter a mão na prática lá no laboratório. Então, eu me preocupo demais em fazer um conteúdo de alta qualidade, sempre pensando lá no longo prazo. Poderia tentar fazer alguma coisa mais chamativa hoje, alguma coisa que desse mais audiência hoje, falar de algum tema mais polêmico, trazer alguma coisa nesse sentido, que com certeza ia dar mais audiência hoje, com certeza absoluta ia dar mais engajamento para o canal, mas eu estou pensando lá na frente, daqui 3, 4, 5 anos, quando eu tiver uma audiência bem maior, eu crescer com as pessoas tendo esse conhecimento que o conteúdo ali é confiável, é de qualidade, assim por diante. Então eu trabalho com muito, muito cuidado. Não é medo, né, de falar alguma coisa errada, já falei coisa errada, normal, é impossível se livrar de falar coisa errada, faz parte do jogo, tem que assumir isso também. A gente que tem formação técnica geralmente tem dificuldade, né? Isso, cara, errar faz parte do jogo. Vai ter coisa errada lá, eu só coloco uma nota informando no vídeo, alguma coisa do tipo para... Se for muito grosseiro, eu tiro, né? Mas geralmente não é. Então, às vezes, coloco uma notinha ali, mas tem que tomar esse cuidado. Eu tomo bastante cuidado com isso mesmo, bastante mesmo. Tanto quando é vídeo para leigo, como vídeo mais técnico.
Interessante. Eu acho que essa questão, se o Frederico quiser complementar, mas essa questão do técnico a gente fica se cobrando muito, né? Então, acho que realmente...
Gente, tem muito
eu tive algumas experiências como professor e aquela coisa de você revisar o conteúdo 1000 vezes, porque você fica naquela cobrança ali do que ser uma conversa mais... Sim, eu concordo com você, não é para você ter menos atenção, mas parece que a sua cobrança é maior. Eu sinto isso, pelo menos no meu universo, na minha bolha, não como divulgador científico, mas com as coisas que eu faço. Frederico, também, se quiser falar, fica à vontade.
Existe também uma questão que, no meu caso, isso acaba acontecendo, que às vezes a gente faz um tutorial para alguma ferramenta ou para alguma abordagem de análise de dados, e aí a ferramenta é atualizada, ou aquele tutorial ele não é mais tão replicável com o tempo, ou por algum motivo da forma como as coisas foram instaladas, funciona no meu computador, mas às vezes uma pessoa vai tentar instalar e não funciona, porque, enfim, tem outro sistema operacional, tem outra versão dos... Então, às vezes é meio aquela coisa que a gente fala muito em pesquisa científica, né? A reprodutibilidade, às vezes, é um pouco difícil, às vezes, né? Ainda mais se a gente começa a botar várias ferramentas ao mesmo tempo, assim, né? Então, isso é uma coisa que eu também acabei tendo que preocupar, assim, nos tutoriais, que é uma coisa que não passava na cabeça antes, assim, né? Tipo... E até alguns dos tutoriais que a gente fez nos últimos tempos, assim, foram para tentar facilitar essa questão da reprodutibilidade, né? Usando ferramentas, assim, que não são nem ferramentas de bioinformática, são ferramentas de boas práticas de programação, né? De engenharia de software, assim, para que seja mais fácil ter essa reprodutibilidade, assim. Então, às vezes acaba... Então, o Omix, assim, teve situações em que a gente teve que trazer ferramenta, fazer uma divulgação não científica de ciência, né, pesada, assim, mas ser uma divulgação científica de outras áreas, né, no caso de engenharia de software, para dentro da bioinformática, assim. Então, isso é também um pouco... uma coisa meio meta, assim, sabe?
Sim. Ó, só vou aproveitar aqui e passar alguns recados, vai ser coisa de 1 minuto, 2, mas lembrando, gente, que depois vai ter lista de presença, então avisem aí seus colegas que estão no evento para entrar, para a gente também conseguir aumentar aí o número de pessoas ao vivo. Então, peço a todos aí depois que compartilhem para todo mundo participar do evento, tá bom? Seguindo as perguntas, pergunta do Josué Luiz Proença Modena. Pessoal, parabéns pelas iniciativas tão importantes. Minha pergunta é relacionada com a do Ricardo, então com a pergunta anterior. Vocês têm se preocupado em como fazer para divulgar conteúdo científico e atrair negacionistas? Gostei da pergunta. Então, essa é uma preocupação no meio de divulgação científica de vocês e como vocês lidam com isso. Natácia, já foi que você foi na ordem a última, agora vou voltar para ti para você iniciar.
Gente, eu não me preocupo, de verdade, porque assim, o impacto que a gente gera é tão maior, é óbvio que, por exemplo, tem alguns temas que eu não vou mentir aqui, eu não gosto de falar. Por exemplo, não gosto de falar sobre evolução. É porque eu não gosto de evolução? Não, é porque é um saco, principalmente no Facebook, a quantidade de comentários que eu tenho lá. Então, algumas vezes confesso que tem uma preguicinha, porque aí, o que acontece? A plataforma ela bloqueia a minha divulgação, porque eu recebo muito dislike, porque a galera não entende o que eu estou falando, eles não entendem que opinião é diferente de teoria. Mas, assim, eu recebo, vou lá, eu respondo algumas vezes, não no intuito de rebater o hater ou o negacionista, mas no intuito de, quando alguém chegar no meu canal, ler o argumento que eu coloquei para ele com muito respeito, e assim, de repente, eu ter uma permeabilidade maior em algumas pessoas que talvez não pensem em alguma coisa parecida e não tenham coragem de se expressar, mas não é algo que eu me preocupe, assim, honestamente.
Eduardo, Frederico, obviamente o público mais especializado talvez seja um pouco diferente do que você está ali, mas eu acredito que sempre há, né? Sempre vai ter ali, em uma porcentagem maior ou menor, sempre há.
Sempre. Eu tenho até uma história engraçada, eu vou contar já. Mas assim, é justamente isso. O fato de a gente falar com pessoas que já são da área facilita um pouco, tem menos negacionista, né? A pessoa já é mais instruída, mas sempre aparece. Faz um ano, mais ou menos, acho que um pouco menos, chegou uma mensagem para mim de uma conta, eu não vou falar nomes, de uma conta verificada no Instagram. Eu fui descobrir que ela era uma esposa de um cantor sertanejo famoso, dos grandes. E ela estava preocupadíssima porque o marido dela estava recebendo mensagens negacionistas, ele estava acreditando naquilo, e ele estava preocupadíssimo com o fato do DNA dele ser alterado por vacinas de RNA mensageiro. Então, ela mandou mensagem para mim, perguntando se de fato tinha algum problema tomar uma vacina de RNA mensageiro, né? Super preocupado, porque ele estava acreditando nisso, não queria tomar a vacina, e mostrou o vídeo de um médico, ela mandou um vídeo para mim de um médico falando da modificação do genoma por vacina de RNA mensageiro, que é um absurdo maluco, né? E eu tive que fazer um trabalho ali, eu me senti obrigado a fazer um trabalho, conversar com ela com bastante cuidado, né? Não só mostrando do ponto de vista técnico, porque com essas pessoas, com as pessoas que estão ali, assim, ó, eu vejo da seguinte maneira, 3 tipos de pessoas negacionistas. A pessoa que de fato tem dúvida sobre aquilo que está acontecendo. Então a pessoa tem medo mesmo do RNA mensageiro alterar o DNA dela, a vacina fazer mal, ela tem aquela dúvida genuína. Essa pessoa você tem que conversar tecnicamente, explicar como é que funciona, ela vai entender, muito possivelmente ela vai entender se você fizer um bom trabalho de comunicação. Segunda, são aquelas pessoas que estão ali acreditando no negacionismo, na Terra é plana, a vacina faz mal, o chip, aquela coisa toda, porque ela quer fazer parte de um grupo, ela quer se sentir privilegiada, ter uma informação que ninguém tem, ela ser diferente da maioria. Com essa pessoa não adianta falar tecnicamente, tem que falar com emoção com ela, tem que pegar na emoção dela. Se você falar tecnicamente, olha esses dados, para ela não vai funcionar. Tem que falar, olha só, tá vendo? Seu filho vai morrer. Você tá vendo o que vai acontecer? Seu pai vai ficar doente. Você não vai conseguir o que você... Então tem que ir por um outro caminho. E a terceira pessoa é mais ou menos parecida com a segunda, só que ela tá ali envolvida politicamente no negócio. E nos dias de hoje tá um negócio maluco. Então, no caso dela, eu senti que era uma dúvida genuína. Era uma dúvida genuína. E foi importante falar. Porque se esse sertanejo, ele vai para as redes sociais depois, falando que faz mal, que ele recebeu um vídeo de um médico e aquilo, pronto. Aí disseminou o negócio de uma amplitude muito maior. Então, assim, sim, aparece pouco, mas quando aparece, fazer o trabalho de contenção de danos aí pode ser importante.
Eu vou até complementar um pouco o que você acabou de falar, Eduardo, que você falou, por exemplo, de um caso do sertanejo famoso, e ontem, na abertura, o nosso tema foi o papel do cientista frente ao obscurantismo na pandemia, né? E eu lembrei exatamente, teve uma fala, acho que foi até o que a Natácia comentou no momento já aqui, que é que essas pessoas, é o funk do Butantan, é o que vai ter ali a larga escala da população, né? Então você vê que, é o que você acabou de falar, essas pessoas que são da mídia são as pessoas que estão mais ali influenciando e como que a ciência chega lá, né? Então, você vê, por exemplo, a audiência que tem um programa igual a Big Brother, se você ter um anúncio ali sobre vacina, a diferença que pode fazer. Então, você falou exatamente disso, achei bem interessante de trazer essa percepção de cientista que a gente não é... Sim, a gente tem que ter esses canais, tanto para leigo quanto para especializados, mas a gente tem que chegar também nessas outras bolhas de alguma forma. Mas eu gostei muito do que você falou. Frederico,
cara,
se quiser, pode
desculpa,
falar.
mas é que acho que vale a pena dar uma puxadinha no negócio aqui.
Pode falar, vamos. A ideia é fazer uma roda de conversa.
Não, é legal, porque vai sair cientista dessa galera que está assistindo aqui. E uma coisa importante, que graças a Deus está mudando com esse negócio de divulgação científica, é que o cientista, ele é um cara que quer sentir, assim como o negacionista, de certo modo, ele quer sentir importante, parte de uma bolha, uma pessoa com conhecimento diferenciado. E muitas vezes, pode pegar aí professor de vocês, pesquisador que vocês conhecem, esse cara, ele não quer se misturar com a massa, ele não quer falar com o povo, porque se ele fizer isso, ele está saindo daquele patamar elevado de importância dele e descendo, entre aspas, né, o nível dele, e aí ele deixa de se sentir importante, de fazer parte da bolha. Então, para a galera que está aqui, não esqueçam disso, cara, que a ciência ela só vai evoluir, você só vai ter um prestígio, a gente só vai conseguir fazer esse negócio crescer se nós conversarmos com todo mundo de igual para igual, sentado olhando no olho de igual para igual, sem palavras difíceis, sem termos técnicos excessivamente, sem querer mostrar que você é especial. A pessoa, ela quer sentir no mesmo nível que você para ela conseguir entender, absorver, aprender alguma coisa que tenha qualidade.
Perfeito, concordo plenamente. Frederico, se quiser complementar, fica à vontade. A gente pode também seguir para as outras perguntas. De novo, a ideia aqui é fazer o mais dinâmico, o mais à vontade possível. O Frederico já está até em casa, o Eduardo está até em casa, está no laboratório. Fazer o mais possível aí, fica à vontade.
Não, assim, até no Omix a gente não fez conteúdos direcionados assim para o pessoal negacionista, mas eu acho que assim, uma coisa que eu tento fazer no limite do possível é uma divulgação científica no dia a dia, né? Tentar quando alguém, né, fala essas coisas que são às vezes para nós que entendemos mais como funciona, por exemplo, uma vacina, por exemplo, ou alguma outra tecnologia, né, ou metodologias de validar, né, um fármaco, né, ao longo dessa etapa clínica, assim, por exemplo. E a gente vê alguém falando uma coisa que tecnicamente é meio absurda, assim, a gente sabe que a pessoa está falando porque não tem instrução, porque acabou sendo induzida por uma... Aí isso a gente tenta, é importante a gente tentar fazer, né, passar o conhecimento de uma forma acessível sem olhar de cima para baixo, né, até porque, na verdade, a gente tem um conhecimento técnico de uma coisa muito específica, aquela pessoa, às vezes, que está falando daquilo, ela também pode ter um conhecimento técnico, um conhecimento prático, às vezes em outra área, às vezes pode ser um engenheiro que não soube filtrar a informação e acabou caindo nisso, né, às vezes pode ser uma pessoa... Claro, às vezes pode ser uma pessoa que não tem uma formação formal também. E às vezes acho que até... E às vezes o que eu acho mais complicado, às vezes pode ser uma pessoa que também é cientista. Porque isso é uma coisa que... Existe cientista negacionista. Existe cientista que, às vezes, por ser de outra área, ou às vezes acaba também. E da mesma forma que às vezes existe gente da área de ciências da vida que pode cair em outras fake news, assim, né? Pode cair em fake news de, sei lá, coisas mais políticas, pode cair em fake news de coisas, sei lá, mudança climática,
Sim,
isso aí existe, né?
Sim.
Então a gente tem, e acho que é importante isso que o Eduardo falou, assim, de a gente sentar, a gente olhar, né, de igual para igual, assim, conversar com uma forma clara, sem ter, né, prepotência, para assim dizer, isso é super importante. Isso é uma coisa que vai além do projeto mesmo, né? Vai além dos nossos projetos de divulgação, uma coisa que a gente tem que adotar no dia a dia mesmo. Vão ter situações em que a gente acaba até... Claro, a gente tem um limite também, né? Tipo, às vezes tem pessoas que estão tão fechadas naquela bolha, assim, né? Que estão tão convictas, assim, daquilo que vai ser um desgaste enorme, assim, para a gente tentar e talvez a gente não consiga. Mas o mais importante, às vezes, a gente dá um contraponto, porque a gente pode explicar coisas, talvez aquela pessoa não mude, mas pessoas que estão na volta, que ainda não estão com as suas convicções, que às vezes não sabem muito bem para qual lado olhar, assim, essas pessoas a gente consegue fazer um papel, né?
Com
Então,
certeza.
acho que é nesse sentido.
Gente, eu preciso falar. Eu
Fica à
preciso.
vontade, a ideia
De verdade.
é realmente virar uma conversa.
Gente, eu
Fica à vontade,
preciso.
Natácia.
Olha, eu concordo muito com o que o Eduardo Frederico falou. O Eduardo tocou num assunto que eu acho assim muito sensível. E, gente, eu queria falar para vocês que é o seguinte: eu coloquei uma imagem de um ouvido com uma lâmpadazinha, justamente porque a gente tem mania de falar demais. Vamos escutar o que a pessoa tem para falar e entrar no mundo, porque geralmente, quando a gente escuta uma coisa um pouco absurda na nossa concepção, e é absurda várias coisas, não estou dizendo que não seja, mas a primeira coisa que a gente fala é: como assim? A pessoa já retrai. Então, vamos escutar o que o outro tem a dizer. E não é que você vai passar a mão na cabeça e vai falar, tá tudo certo. Não, mas por que ele está pensando dessa forma? Deixa eu entrar dentro do universo dele para eu conseguir entender o contexto que ele está dizendo e por que ele está dizendo. Porque senão a gente cria muitas barreiras, tá? Eu costumo dizer, por exemplo, quando eu estou fazendo divulgação científica para crianças, o que eu faço? Eu agacho e eu olho no olho dela. Entendeu? Nem acima, nem abaixo. Eu agacho na altura dos olhos e converso, porque eu não sou menor nem maior, eu sou igual a ela. Então, eu acho que a gente tem que fazer isso para a gente conseguir ter essa permeabilidade maior na sociedade, sabe? Às vezes eu vou falar dentro do meu canal, por exemplo, de haplótipos, e eu uso donuts como exemplo. Porque eu me formei em São Paulo, fiz meu doutorado aí, então a gente esquece que aí a concentração de informação é muito grande, mas a gente esquece que não necessariamente isso é a mesma coisa num estado, por exemplo, no interior de Goiás, ou interior de outros, estou falando no meu contexto, né, de outras localidades no Brasil que é tão grande e é tão heterogêneo. Então, a gente precisa exercitar a paciência, ouvir e conversar, tá bom?
Com certeza, concordo. Obrigado, Natácia,
Imagina.
pelo complemento. Vou pôr mais uma pergunta? Quais são as dificuldades que vocês enfrentam no dia a dia ao fazer divulgação científica? Gostei, né? Não é só as mil maravilhas, também falar um pouco aí da dificuldade dessa profissão aí, que vocês acham que são do dia a dia, quais são as dificuldades para cada um. Eduardo, fica à vontade para começar.
Vamos lá. Primeira coisa, é bonito, né? Ver o youtuber lá, o divulgador científico. É legal a gente ver aquela pessoa exposta nas redes, com fãs e tudo. Não é o meu caso, não. Tô falando tipo o Serjão, do Space Today, esse pessoal grande, sabe? Só que a gente tende a romantizar essa situação, esse ambiente que eles vivem. Fazer divulgação científica, trabalhar na internet é um negócio que demanda tempo. Tanto que eu não faço absolutamente nada da vida a não ser isso. Eu vivo disso aqui. É isso aqui. Esse é meu ganha-pão. É assim que eu sustento minha família. É com isso. Então, hoje, por exemplo, eu acordei 10 para 6 da manhã. Já comecei a estudar a aula que eu tenho que preparar para hoje à noite. Eu não está 100% pronta ainda. Vou ficar o dia inteiro fazendo isso. Vou até as 9 da noite dando essa aula, tirando dúvida do pessoal. E amanhã começa de novo. Além de ter que estudar tecnicamente, preparar o conteúdo técnico que é o que aparece, tem todo o backstage aqui que me permite fazer isso. Então eu tenho que aprender a enviar e-mail, porque enviar e-mail, né, quando você envia para uma pessoa é uma coisa, quando você envia e-mail para 27 mil pessoas, é diferente. Então tem que saber usar a ferramenta do jeito certo. Tem que aprender a utilizar o YouTube de um jeito profissional, tá? Tecnicamente falando, né? Usar os analytics, identificar onde estão os furos dos vídeos, o que tem que dar. Tem que aprender a fazer anúncios, porque tem que anunciar, não adianta tentar crescer só no orgânico, que isso aí é para poucos, é para quem viraliza. Tem que aprender a fazer anúncio, que é um negócio difícil, toma bastante tempo. Eu tenho hoje já um funcionário para me ajudar, então eu tenho que cuidar dele, ele tem que fazer as atividades dele do dia a dia. Tem a empresa como todo, porque o Universo da Biologia virou uma empresa com CNPJ, que emite nota, tem toda a burocracia envolvida de uma empresa. Então, a maior dificuldade da divulgação científica profissional não é a criação do conteúdo, é todo o resto que está ali para dar suporte à criação do conteúdo. Tudo isso que eu falei, da empresa, o funcionário, tudo. É o conjunto das coisas e aprender coisas que você nunca imaginou que precisaria aprender para conseguir fazer esse negócio funcionar. Essa é a minha maior dificuldade.
Legal. Gostei de conhecer um pouco esse universo. Frederico, fica à vontade para complementar também. Suas dificuldades no dia a dia.
Acho que as principais dificuldades que eu tive assim no começo, principalmente, foi filtrar o que eram temas que eu gostava muito de falar, mas que, tipo, não tinham, talvez, que estavam, talvez, muito à frente, sabe, do que talvez as pessoas estivessem procurando, assim, que fossem coisas muito específicas que talvez não tivessem tanto uma... Porque, para mim, assim, se eu pensei, bom, posso começar aqui a falar sobre, sei lá, análise de pangenomas, por exemplo, ou alguma outra coisa assim, alguma coisa muito específica de sequenciamento de nova geração, por exemplo. Só que, tá, isso é uma coisa que eu quero falar, mas talvez não seja isso que as pessoas estejam tão interessadas, ou talvez, tipo, elas queiram algo antes, assim, sabe? Elas queiram talvez entender antes, tipo, plataformas de sequenciamento de nova geração, entender por cima alguma outra aplicação para depois poder chegar em montagem, para depois poder chegar em esse outro tópico mais avançado, né? Então, assim, eu acho que uma dificuldade que eu tive no começo foi, tipo, como, sabe, construir conteúdos que, tipo, gradualmente fossem levando as pessoas para entender essas coisas que eu achava, assim, mais legais de falar, assim, porque, enfim, era minha área de pesquisa, né? E, tipo, saber, às vezes eu me esqueço, assim, tipo, o tempo a gente acaba esquecendo, né? O quanto no começo, algumas das coisas que depois vão se tornando triviais, no começo elas são mais difíceis. Tipo, às vezes, tipo, sei lá, como no começo, assim, alguns colegas de graduação tinham dificuldade de entender o BLAST, por exemplo, ou algumas outras ferramentas mais triviais, assim, de bioinformática, sabe? Então, uma coisa que ajudou foi que o Omix ele surgiu como um projeto pessoal, ele surgiu depois que eu terminei o doutorado, e eu estava fora da academia e eu queria ter uma forma, tipo, de também além de querer divulgar conhecimento, né, e querer de retribuir, eu queria também ter uma forma de me manter conectado à área, assim, manter refrescando, assim, sabe, isso na minha cabeça e poder passar isso para outras pessoas, para que esse conhecimento fosse útil para outras pessoas. Então, assim, depois eu voltei para a academia, depois de um ano e pouco fora, quase 2 anos, e aí uma coisa que ajudou, assim, a manter, assim, essa ideia, né, de o que as pessoas estão precisando é incorporar mais gente na equipe, assim, incorporar pessoas de graduação que estão começando a estudar bioinformática, e incorporar pessoas de pós-graduação que já têm um conhecimento de alguns conceitos, mas estão usando nos seus projetos e vão sentindo algumas demandas, sabe? Então, assim, a dificuldade às vezes é, tipo, manter aquilo que está sendo produzido próximo daquilo que tem uma certa procura. E, claro, tipo, o Instagram também ajuda, né? Tipo, a gente poder fazer, às vezes, perguntas, assim, tipo, e entender melhor o que o público está precisando, assim, né? Então, isso...
Legal.
E fazer, às vezes, também... vamos fazer um quiz aqui para ver se o pessoal está familiarizado com esses conceitos, né? Tipo, você sabe o que é microbioma, você sabe o que é NGS, sabe o que é uma OTO. Não, tá, beleza, as pessoas não estão sabendo bem o que são essas coisas, então vamos, de repente, fazer um post falando sobre isso, porque tem uma procura, né? Então, acho
Bem
que nesse
interessante.
sentido...
Gostei também. É interessante a gente ver de fora, né? Pelo menos eu acho, até antes de passar a palavra para a Natácia, é muito bom, porque a gente vive hoje num mundo de bastante divulgação científica, acho que ainda mais depois da pandemia, obviamente que é um contato mais para leigo, mas quando eu imaginei que eu ia ver minha mãe falando PCR, né? Então acho que a gente vive hoje no mundo sobre muito mais contato com divulgação científica, mas ter a ideia dos problemas dessa profissão, porque é uma profissão, que nem o Eduardo falou, você vive disso, é uma profissão, também é muito bom para a gente ter aí, e quem sabe servir de incentivo para outros pesquisadores aqui do grupo desse evento serem também divulgadores científicos. Natácia, as suas dificuldades aí do dia a dia como divulgadora?
Gente, eu vou dividir em 3, tá? A primeira foi quando eu comecei. A maior dificuldade foi começar mesmo. Tipo assim, falar, vou fazer. Esse passo foi difícil, porque eu pensei, por que vão querer me ouvir falar se tem tanta gente falando? E a gente esquece que, às vezes, a pessoa se conecta com a sua forma de expressar, com a sua forma de pensar. Então, tem espaço para todo mundo. A segunda foi, meu Deus, o que eu vou falar? Então, definir alguma coisa específica. Por exemplo, muita gente chega para mim perguntando questões de biologia molecular. Eu já encaminho para o Eduardo. eu confio no conteúdo que ele produz, então eu falo, a gente vai para lá, porque eu não vou falar disso. Eu não vou falar aqui, eu vou falar sobre genética de populações, genoma populacional, flogeografia, ok. E a terceira é entender que feito é melhor que perfeito. Porque a gente vai errar, vai dar ruim. Por exemplo, você está me ouvindo falar aqui, pensa, nossa, é fácil para ela falar. Não, não é fácil para eu falar, entendeu? Não foi fácil fazer a primeira live, não é fácil abrir stories, e as pessoas cobram que você esteja ali. Demanda muita energia, porque eu não vou nem repetir o que o Eduardo falou. Quando você tem um CNPJ, é tudo o que ele falou. A gente aprende a fazer marketing, tem que estudar posicionamento, tem que aprender sobre estratégia, você tem que entender como que funciona um e-mail marketing. Então, no nosso caso, que nós somos divulgadores profissionais, existe um background, por exemplo, emissão de nota fiscal. Vocês acham que eu sabia que era isso? Sim, isso agora faz parte do meu universo, né? Então, isso tudo eu acho que hoje, atualmente, é você conseguir equilibrar todos esses pratos e fazer uma boa divulgação científica, porque o que a pessoa olha no YouTube é só, assim, a pontinha do iceberg que tem por trás para tudo aquilo ali estar disponível, tá bom? Acho que esse é o meu maior desafio hoje, né? Equilibrar todos esses pratos que tem aí por trás do backstage.
É um grande backstage, deu para ver aí bastante que é um grande backstage. Gente, são 9h26, a gente infelizmente está chegando aí no final. A gente colocou aqui, a comissão colocou nos comentários o Instagram aí das páginas, então eu peço encarecidamente para todo mundo que está ouvindo aumentar aí esse número de seguidores, assistir, compartilha. E é engraçado, né? Que nem você falou assim, não é difícil, eu gostei da frase da Natácia agora, não é difícil, não é fácil você estar ali falando, né? E realmente, é que nem eu fico aqui mega nervoso só por estar presente aqui, tá falando já é uma dificuldade, né? Às vezes as pessoas, não, você fala super bem, fala assim, você não sabe o quanto minha perna tá tremendo. Não é tão fácil assim também quanto às vezes parece. Eu vou passar a palavra para vocês encerrarem, então, para cada um aí falar suas palavras finais, considerações, para depois finalizar. Agradeço de novo a presença de cada um, foi uma conversa ótima. Espero que vocês também tenham gostado, tá? Fica o agradecimento aí de toda a comissão. E vou começar com o Frederico, então, para essas palavras finais e a gente finalizar essa mesa redonda e agradeço de novo, tá? Frederico, a palavra é sua. O senhor está no mudo. Um minutinho.
Eu acho que a divulgação científica ela não é uma coisa que deve ser uma obrigação de todo cientista, mas é uma coisa que tem que ser pelo menos estimulada, sabe? Para toda pessoa que está numa formação relacionada à ciência. Assim, acho que nem todo mundo tem, às vezes, inclinação para isso, ou vai se sentir confortável com isso, mas é uma coisa que a gente tem que pelo menos tentar exercitar, assim. E acho que o melhor exemplo disso é esse filme que saiu há pouco tempo na Netflix, o Não Olhe para Cima, porque ele tem o tapa na cara do negacionismo, mas ele tem o tapa na cara, às vezes, da incapacidade dos cientistas de se comunicar. E acho que ele é uma coisa que a gente tem que olhar ele também como uma crítica não só à sociedade, mas à academia, que às vezes até se vê distante da sociedade, sabe? Às vezes fica no paláciozinho de cristal, às vezes não olha assim, fala com a linguagem, né, das outras pessoas, né, mais leigas nos assuntos, assim. Então, acho que a divulgação é uma coisa super importante que a gente tem que exercitar em quem está em formação acadêmica, uma soft skill super importante, assim, até para a ciência ela passar a ser mais valorizada. É muito mais fácil, a gente certamente vai conseguir ser melhor visto como cientista pelas outras pessoas, né, pela sociedade de modo geral, se a gente conseguir falar o que a gente pensa, o que a gente descobriu, o que a gente precisa de uma forma mais clara.
Perfeito. Muito obrigado, Frederico. Eduardo?
Boa, foi um bom fechamento, Frederico. É isso aí, cara.
Também gostei.
Não, eu acho que é fantástico, cara, porque é bem isso mesmo. A gente tem que falar, conseguir se comunicar com as pessoas para a gente conseguir divulgar, né? Para as pessoas entenderem a importância da ciência, que tudo que está aí em volta delas, de certo modo, tem o dedo da ciência, né? Então, isso é fundamental, sair do pedestal e se comunicar de igual para igual. É isso aí. Ô, Ciro, eu posso fazer um jabá de 1 minuto?
Claro, fica à vontade.
Para o pessoal, eu vi que já colocaram aqui o link das redes sociais, mas só para ficar claro, ó, Universo da Biologia Molecular no YouTube, o canal do YouTube, ali tem aulas completas sobre biologia molecular. Então, ali é para quem quer se aprofundar nas técnicas, entender como é que funciona, quem vai utilizar isso no dia a dia mesmo, ali as aulas são mais profundas. No Instagram, que está aí, né, o @Universo da Biomol, Ali a conversa é mais direta, um papo mais direto, conteúdo mais direto ao ponto, mais pessoal também, que eu abro as caixinhas de perguntas e respostas, pode entrar em contato comigo diretamente. Então, o conteúdo, de um bom sentido, mais superficial, mais rápido. E, para finalizar, quem quer dar um passo a mais, quem quer aprender biologia molecular mesmo, se profissionalizar nisso, eu tenho uma escola de biologia molecular, que é o Clube do DNA. O Clube do DNA é uma escola de biologia molecular, que tem aula toda semana, tem curso completo lá, ele funciona por assinatura, estilo Netflix, e é baratinho, conteúdo excelente, com toda a modéstia de lado, mas conteúdo excelente, com apostila, com certificado, com nota fiscal, com tudo certinho lá, tá lá, beleza? Se você quiser conhecer o Clube do DNA, é universodabiologia.com/cdna. Beleza? Obrigado pela oportunidade, valeu demais, Ciro, valeu pessoal da organização, é isso aí.
Imagina, a gente que agradece. Natácia, fica à vontade também.
Gente, muito obrigada pelo convite. Eu quero dizer para você que a comunicação, ela permeia todas as nossas relações. Então, às vezes a gente tem mania de separar as coisas em caixinha, né? Divulgação científica, comunicação. Mas a verdade é que quando a gente comunica, né? E eu digo para você que eu cresci muito do ponto de vista de comunicação, de fala, de expressão, quando eu abri o meu canal. Então, a gente tem que parar de separar em caixinhas e entender que a forma de você falar com seu pai, a forma de você falar com a sua mãe é uma forma de divulgação, é uma forma de conversa. Então, não precisa separar, não precisa ter esse medo. Eu estimulo todos vocês a terem essa experiência. A gente sabe que dentro da universidade você precisa, se não mudou, ter 10% de carga horária aí dentro do seu curso de extensão. Então, não tem para onde correr, a gente tem que beber um pouquinho dessa fonte. Eu trouxe para vocês diferentes exemplos, porque eu sei que nem todo mundo vai querer ir para a internet, ou nem todo mundo vai ter, sei lá, um perfil que se alinhe com um público jovem, infantil, enfim. Cada um tem aí a sua, digamos assim, a sua inclinação. Então, eu desejo para vocês sorte. Se vocês tiverem alguma dúvida, pode entrar em contato comigo lá pelo Entenda Genética. Vai ser um prazer. E, vão divulgar ciência. Abra um perfil, começa agora. Eu sempre vou estimular isso, estimulo todos os meus amigos a entrarem nessa jornada, tá bom? Muito obrigada a todos vocês.
Agradeço de novo aí, foi uma ótima conversa. Natácia, Eduardo, Frederico, obrigado de novo. Tenham uma ótima semana e fiquem à vontade para acompanhar o evento aí no YouTube, tá bom? Obrigado de novo. Bom dia.
Valeu, gente. Bom dia. Tchau, tchau.
Valeu, pessoal.
Bem, pronto, tá certinho. Dando continuidade, a gente vai agora passar para as próximas palestras. Alguns recados a gente falou ontem, vou falar hoje de novo. Como a gente tem alguns palestrantes internacionais e a ideia é que esse evento seja sempre o mais internacional possível, que atinja outros públicos, a gente vai algumas vezes mudar de idioma, então sempre é bom reforçar isso. As mesas redondas, no caso, a gente sempre vai fazer em português para que seja algo mais fluido, mas a partir do momento que a gente passa aí para algumas palestras, vai ter essa mudança de idioma, então eu sempre gosto de reforçar, para não ficar tão estranho do nada essa mudança. Eu vou chamar, então, agora outro membro aqui da comissão, o Márcio, para ele dar continuidade. Qualquer dúvida, gente, fiquem à vontade, só colocar aqui nos comentários. Obrigado, bom evento. Bem-vindo, Márcio. Está me ouvindo? Só para a gente testar o som, tudo. Você ainda está no mudo.
Está me ouvindo agora?
Agora sim, agora está perfeito. Então, fique à vontade, Márcio, pode deixar que a gente está aqui no backstage para colocar o palestrante.
Okay, thank you, Ciro. So our next guest is Professor Philippe Cabrero. He started his academic career as biochemist at Paris Diderot University and did his postdoc studies in the UK at the University College London, where he set up his independent lab in 2013. In 2018, he took up a readership and MRC investigator position at Imperial College London. And in 2021, he joined the Cluster of Excellence in Aging Research at the University of Cologne as professor for microbiota in aging. By combining classical and advanced microbial genetics, high-throughput genomic, and chemical approaches, Professor Cabrero studies both host and microbial physiology in very fine mechanistic detail. Dr. Cabrero, thank you very much for joining us.
Can everyone see my screen? I'm going to go now into full mode, and I hope that will still work. Okay, can you still see me? Is everything working all right? Because right now I only have my presentation in front of me.
Yeah.
Okay, so here's the pointer. So as Marcia said, and very well, so we're interested in understanding really how microbes can regulate host physiology. And what you can actually see here is actually a nematode worm called C. elegans. And you see very nicely here, actually, in almost a surreal sort of painting, the different molecules that would potentially be used as the ways to communicate between these microbes and the host cells. So today I will guide you through different works that we've done in the lab and explain why we use different models to study these interactions. So I don't think we need to go too much into the knowledge about the microbiome. I think it's pretty much common knowledge now that the microbiome is important for human health. What is actually very striking is just by looking at number of genes, you see that our genome is actually very, very small comparatively to the number of the catalog of genes that you would find in the microbiome. So it's almost 100 times greater, the number of genes in the microbiome. And the beauty of that is, therefore, we have this extra organ with so many genes that we can actually potentially change with more ease than we can actually do with a human genome. But what I really want to stress about this is actually the two kind of work together. And a concept that was coined a few years ago by the Rosenbergs is the concept of the holobiont. That we're not just host cells and we're not just bacterial cells, we're actually a combination of the two working together to shape this unit called the holobiont.
Professor Cabrero?
Yes?
Excuse me. We are not seeing your presentation in full screen, I think.
Oh, okay. So can you see it this way?
Yes.
Okay.
But
then I'll
it's
see
not
it this way
on.
probably. Yeah, okay. So the previous slide, yeah, it was just this, so it's just a couple of diagrams and images. So there's been a lot of research now, and this is just a snapshot of 3 key papers where you can actually see that this microbiome can really be changed by the things that we take every day, whether it's the diet that we eat or the drugs that we take in a regular basis to treat different kinds of diseases that we start having as we get older. And they all seem to have a dominant impact on shaping this microbiome to a degree that it's even more important than actually our own host genes in shaping the microbes themselves. So therefore, this environment really, really has the ability to change this flexible organ. So the areas of interest of our lab are kind of focused on these 3 main topics. Aging is one of them, and cancer is another. And in general, really, xenobiotic or drug metabolism is something that it's quite close to my heart in the sense that it's something I studied already at university, and I kept being interested even throughout my entire career. And overall, over the past 10 years, there's been kind of 4 main mechanisms by which we can already understand how drugs shape physiology, and this is or drugs or even just bacteria can shape bacterial physiology. So the first one would be that bacteria can transform drugs that we take. A second one, that bacteria can regulate the mechanisms on which these drugs can work on. Bacteria can change their own metabolism and their signaling pathways and metabolic pathways by drugs or even diet or even a combination of both. And something we're still working on nowadays is that the microbiome can actually develop resistance to drugs that we naturally take. We know that antimicrobial resistance is one of the biggest challenges for humans in the future. And what we're realizing is that not only the bugs develop resistance to classic antibiotics, but they also seem to be acquiring new mechanisms of resistance when we're talking about drugs that we naturally take just to treat disease. So the first one of our key papers was in 2017, and we're able to show that actually microbes have the ability to activate cancer drugs. So a lot of the drugs that we take normally for treating cancer are what we call prodrugs. So they, on their own, they don't have a bioactivity, so they need some sort of activation. For a very long time, we thought that that activation was only being done by the liver, but we're now realizing that several microbes have the ability to also activate these drugs and make it more effective. I will show you the work that we did in order to show these important mechanisms. Another mechanism is that drugs may actually be buffered by microbes. So the microbes will take these drugs and then they become less available for efficacy on the host. This was work done in collaboration with other groups here in Germany, and I'll discuss this also briefly today. The second mechanism is really was showing, and this is work in progress, and I won't be talking about this today. Hopefully this or next year, work will be published on this, but we're showing that indeed microbes also have the ability to produce metabolites that can have an impact on the cells of the host, making the drugs that we take more or less effective. And this is really important because it's something quite overlooked, is that microbes have the ability not only to transform these drugs, but actually produce other things that can influence physiology. And in a similar sort of line of work, in 2020, we've shown that actually microbial metabolites produced by bacteria could influence longevity and health span of a host. The work that I'll be also discussing today is a follow-up from our original work in 2013, and now in 2017, we've shown that certain drugs like metformin can rewire bacterial metabolism in a way that it makes them produce certain metabolites that have positive effects on host physiology. And you can see here we use different models to show these effects. We use the C. elegans, the fly, and also we did some human computational studies. And finally, the work that I will not be talking is that indeed microbes can develop resistance to certain drugs, and this developing of resistance actually reduces the ability of these drugs to work, either through the production of metabolites or through new mechanisms by which the drug is less available to the host. And this is being done, again, in C. elegans, in mouse models, but also in human samples. So the microbiome, this is a summary of basically what I just said, indeed, and this is a very nice review if you're interested in this topic. And indeed, it does have this incredible metabolic potential to really alter how drugs work. And the way it does that is either through direct or indirect mechanisms. But how do you study really host and microbes together when you combine? So hosts are already complicated. We have different organs, different types of cells. Organs communicate between each other. If you add the layer of that where you have now microbes communicating with the host with a constant exchange of metabolites where both of them are under a feedback loop of regulation, the moment you start adding drugs that can influence either one or the other, or nutrients that can influence one or the other, it becomes a very complex scenario. So how do you go to study in fine detail these very complex interactions? So this is why we use the C. elegans as a starting point, because it really allows us to break down this complexity. We can, for example, study microbial single species effects on host physiology. For that, we can use gain of function libraries, loss of function libraries, where each one of the genes, for example, in a particular bug has been deleted. So we can really go to the fine molecular mechanism by which these microbes can regulate physiology. We can work in complex communities too, using C. elegans, where different microbes can be changed and modified to study how that influences the stability of the community and how that affects the host. A lot of this work is then complemented by computational work. We can then change and also start adding interventions. We can use drug libraries. Here are a couple of examples: the LOPAC, the Prestic library. Diet, we have ways to study individual nutrients on how they affect the physiology of the communities or specific microbes. And we can even design microbes. We can change their function by molecular biology techniques to really study how these influence the abundance of the communities or even the function of their own microbes and how these produce metabolites of interest. Of course, we use the C. elegans because it's a great tool for two things. One of them is their genetic tools. We have it's easily we can easily create mutants using CRISPR-Cas. We can do foreign reverse genetic screens, which is very easy to do, and we even do tissue-specific transgenesis, really allowing us to study how different tissues can respond to specific cues. We can also and this is really why I like to use the worm, is the ability to study phenotypic readouts at a very deep phenotype level. We can study in a high throughput manner many physiological readouts, but also molecular readouts. And this is really something that only the worm can do that. Of course, you can add a layer of that to use the techniques that most labs now have at their display, and this can be done both for C. elegans and microbes. But really the key is then to use that information to move on to other organisms like flies, mice, and humans, or even sometimes the reverse translation, where we find something in a model like this, and we try to work out the mechanism working backwards. So this has been very nicely reviewed in a study, in a review paper from last year. So the first one I'm going to talk about is really about cancer drugs and their role in cancer. This was the work done by 4 very talented people in the lab, and it really is known now, and we're already in 2022, so this is becoming a bit of an old review, including one that we wrote, but it's really well known that microbes, by very different mechanisms, can either increase chemotherapy or reduce chemotherapy. And equally, they can also improve immunotherapy and reduce immunotherapy, although we're not going to talk about this at all today. So fluoropyrimidines are drugs that have been around for almost 60 years now. They are pretty much in every single cabinet in hospitals in the world because it's a drug that is easy to administer. It's quite cheap. But there's something that for a long time people didn't quite understand, is why there were such high attrition rates in patients. Why did some really respond very well to the drugs while others don't respond at all? And that could be potential to host genetics, although for 30 years of studying into host genetics, we couldn't find a very strong association with one gene and efficacy of these drugs, suggesting that maybe something else was playing a key role. And of course, at that point, microbiome became an important factor to be studied, but more and more now also diet is becoming potentially the thing that could explain, in combination with the microbiome, why certain drugs work well or not. And it was also very well documented that people to take these drugs develop a gastrointestinal upset, but it was not clear also with patients that have colorectal cancer whether you would have the changes in the microbiome that were observed were due to the treatments themselves or to the disease. And now the scenario seems to be it's a bit of both. So what was really the role of the microbiome in fluoropyrimidine action? And this was a question we started asking in 2014. And again, this slide here is just really to show why we use the worm, so to study these processes. Because when these drugs are taken by humans, they need the liver and the enzymes of the liver to activate these drugs, and they use these pyrimidine pathway in order to achieve that. So the drug is given as a prodrug, and it becomes activated into 5-FuMP and eventually FDUMP, which are inhibiting an enzyme called thymidylate synthase. And this enzyme is important for creating the nucleotides, which are really the key molecules that allow cells to divide. And what you can see very quickly is that all of these enzymes that we see that exist in the human cells have a very clear functional homologue in C. elegans. So we can really use the worm to start addressing these key questions. And this is just a small quote from Friedrich Nietzsche, a philosopher, who said, "You've made your way from worm to man, and much within you is still worm." And I think this really captures why we use C. elegans to study human processes. So the beauty of worms is really the ability to do very simple assays that can be very meaningful. And one of the questions we asked at the moment, where we just placed C. elegans to produce a progeny, they produce eggs, and these eggs, like human cells, need to divide in order to become another developed organism. But these drugs stop that cellular division. So we can basically place worms in a plate with drugs and then see what is the concentration that you need to stop these eggs from developing into an adult. So we get a very accurate measure of that. And we can then change the type of bacteria that these worms are being colonized with. So what we see is that if you, for example, you place the worms with a bacteria called E. coli B, you need about 4 micromolar of this drug to completely arrest the development of these eggs. Instead, if you place them in another type of E. coli, W3310, you actually need 0.1. So you need 40 times less the amount of drug to obtain the exact effect on the host cells. So clearly the bacteria are doing something to these drugs. We don't know yet. At this point, we didn't know exactly what they were doing, but we knew immediately that bacteria could completely change the efficacy of these drugs, and by a mechanism that we needed to understand. And this is what we moved on to forward. So we performed another very simple experiment. Was it just bacterial activity, or was it something more like a dietary component of these bacteria that could be influencing the effects? And in order to do that, we just destroyed that bacterial activity by irradiating by UV or by heat killing the bacteria. What you see now is that if you have the bacteria alive, you need 0.25 micromolar, but if you destroy the biological activity of these bacteria, you need now 32 micromolar, which is 160 times more. So we really knew at this point, we were fully convinced that bacterial activity had a key role in playing these effects. But we didn't know whether they were activating or deactivating the drug, whether they were producing metabolites that were influencing this mechanism. So we didn't know. So we had to come up with something to understand that. And this is where we developed our first chemical genomic host screen, which had never been done before. So we call it more simply a 3-way screen. Historically, microbiologists have looked how a drug, for example, can influence the bacterial growth of a bacteria when you knock down or knock out each gene individually. So some of these genes will make the drug work better in terms of destroying this bacteria or making it grow less, and some of these genes could make it even work better. So we wanted to know whether we could just simply, by adding the host as a readout of these interaction, was sufficient to reveal function. And we measured, and this is the beauty of the worm, we measured 55,000 phenotypes. And how can we do that? How can we measure such a big number of phenotypes? This is done by visual inspection, for example, in this clear case. So if you put worms on a well without any drug, they develop into these big adult worms you can see here, and they start laying eggs. But if you put drug, 1 micromolar or 5FU, you can see that the worms really struggle to develop. So it's almost a binary information. Yes, it does, yes, or it doesn't. Here is just an example of one of those mutants. If you put them now with these mutants, you can see that the worms on the control develop. The number of worms really doesn't matter. It's just really whether they can develop or not. And if you put them now with the drug, you can see that the worms are developing as well as without any drug, and much better, of course, than when the bacteria was wild-type. So we can now ask the question, at which concentration this mutant will become like this? And this gives us a measure of the contribution of each gene in bacteria that can have a role in the effect of a drug. So this becomes complex data. And what we did, so this is the entire Kaya library. The Kaya library has 4,000 different bacterial mutants. Each one of the mutants was tested. And we plotted this in a 3-dimensional way, which can be quite complicated. So I'll drive you. So what you see on the x-axis is the bacterial growth. So each point here, so this bacteria here would grow a lot better than this bacteria here. This is in the presence of the drug. Again, this bacteria here, this mutant will grow better in the presence of the drug than this bacterial mutant here. But what we really wanted to know, and what you expect, is most genes don't have an effect, and things that deviate from the trend line will either act antagonistically or synergistically. And what you see now is the color. So on the color, you see a gradient going from black to red. And if the expectations, according to our slide where we said, well, can we learn anything from doing this three-way screen that is not recapitulated in just the way people normally used to do, which is this X and Y of drug without drug bacterial growth. So if indeed the worm was just capturing what these assays were normally doing, we would see a gradient in color going from in this diagonal or from bottom to top or from left to right, depending on which variable they would be responding to, but we didn't see that. So that told us that the worm was capable of capturing evidence that was not recapitulated by the normal way that historically people have been doing. What we can do with that is that each one of the genes can be placed into a metabolic pathway, and we can start doing enrichment analysis to see if something comes up that is more than a single gene. And we could see that, for example, the vitamin B6 pathway was very strongly captured. So not only the coverage, which is represented by a color gradient from a purple to a light blue, which shows that more of those genes came up as being involved in the process that we're studying. But also we found different folate metabolism and energy metabolism. So we can go on and then combine all that information to work out exactly the mechanisms that we did. And here we're going to jump that, but basically we found that vitamin B6 and B9 were regulating an important pathway in bacteria through flux, metabolic flux, and this changed how much of the drug was being taken by bacteria and being activated by these enzymes here, UPP, UDP, UDK. We see that there was a redundancy between these enzymes, which had not been shown before. And we then perform metabolomic or mass spec assays to measure the activation of the drug both in bacteria and the worms, and this is what you see here in the worms. So if you put worms on a control bacteria, you can see that there's a big abundance of the activated metabolites here. This is the controlled metabolite. And when you put the worms on bacteria that cannot activate the drug, you can see that the drug completely disappears. So we do not see any activated effects. We went on to really test this in a cancer model of C. elegans. Basically, what we see is these worms naturally, their cells from the germline just continually develop, eventually fusing the two gonads. And when you give them the drug, you can see this very nice separation of the two gonad arms. But if you now give the drug, but you give them on these different mutants that can't activate the drug, you can see that the germline is still fusing. And this translates into a physiological effect because these worms are short-lived. When you give them the drug that really stops this fusion of the gonad, you extend their survival. But if you give them mutants that can't activate, respectively, 5FU or 5FO, you can see that that effect is particularly suppressed. So this was an important paper, I think 2017, where we show that the interaction between drugs, microbes, and hosts can't just be studied individually. You need to study all factors together. That microbes regulate this mechanism through the pathways that I mentioned, vitamin B6 and B9 or folate, and that this activates the drug into making it the efficacy higher on host effects. We also discovered another pathway at the time, which I'm not going to discuss too much, and I recommend, if you're interested, really to read the paper. So, but can we do something? Can we start playing with microbes in order to make them work better? And we did this as a proof of principle. So if we take this bacteria that doesn't activate the drug well at all, and taking the information that we discover in this paper, we can actually make it better. So if we knock down a gene called NDK, we can already improve this effect by 4-fold. If we combine this effect with another gene, YGJJ, which again, this is all to do with rewiring metabolism. Now that we know what are the genes that make it worse and make it better, we can play with them. And if you combine the two together, you can see now an improvement of 40-fold. And these then translate into a tumor retraction. So there's a much greater effect if you combine the drug with these bugs that don't have these two genes. and this also translates into a very strong increase in survival by adding the drug in combination with these bugs. So if, let's say, if a microbiome of a person didn't have microbes that could do this effect correctly, we could potentially change those microbes to make these drugs work, as well as someone who would have a more efficacy-prone microbiome. So the second story of today is really about metformin and longevity. This is something I've been working almost for 14 years now. And metformin is one of those drugs that a lot of people with type 2 diabetes are on. They're taking it on a daily basis, and they will take it for a very long time. And in fact, with the exponential growth in people with type 2 diabetes in Western societies, and the amount of metformin that people need to take a day, which can be up to 2 grams of compound, we can actually start now detecting metformin in rivers. So if you take samples of rivers from all over the world, especially in westernized societies, you can actually detect metformin in rivers, which is quite a scary thought. But one thing that was quite striking about this drug, again, with the pursuit of drugs that can improve longevity, is finding drugs that can mimic dietary restriction. Dietary restriction is known to extend longevity in a variety of organisms, but finding drugs that can mimic that is a very different story. And historically, metformin seems to suit that position a little bit. And one of the key studies that pushed that people to study metformin is this meta-study in the UKPDS cohort, where you could see that survival over time of normal people without metformin, people with sulfonylurea, which is another type 2 diabetic drug, were living shorter than people that had type 2 diabetes on metformin. So what I want to really say about this is people who have a disease but being managed with metformin were living longer, surviving for longer than even healthier patients without any drug. And that was quite a surprising factor. So that led to a lot of work. And more recently, people focused on how, especially from our study in 2013, that how does really metformin work with the microbes? We know that people on metformin have very important gastrointestinal upset, but really mechanistically what was going on was not clear. There's many mechanisms that people have been describing. Again, metformin historically has been associated with many different mechanisms too in the host cells, and similarly now we're finding metformin seems to act in very different ways with different microbes. But one thing that seems to be quite consistent throughout both animal model studies but also human studies is that metformin seems to increase the abundance of Escherichia coli. But the exact mechanisms of that were not until very well known until the work that we did and some others. So we took that from that point on, and what we did was to give metformin to mice on a normal diet or a high-fat diet. And you can see that if you give mice a high-fat diet, they gain more weight over time compared to a normal diet. And if you treat them with metformin, you can suppress this gain weight. We now analyzed both the lean and the fat mass of mice, and we saw that this increase in fat mass, the weight that we saw, was specifically associated with the fat mass. We didn't see any changes in lean mass, and this effect was particularly suppressed in the metformin supplementation. We then went on to give mice, we took germ-free mice that are devoid of any microbes, and either gave them metformin or colonized them with E. coli. We colonized with other microbes, and what we saw was actually very striking. So microbes that are given to, the first point that was very interesting was that when you colonize a mouse and you give them a high-fat diet, they gain immediately a lot more weight than the germ-free animals. This is been known, but it's nice to see that replicated here. But what we saw was that if you are a germ-free mouse and you give them metformin on a high-fat diet, that makes no difference. But if these are colonized now with E. coli and they gain more weight, this effect was suppressed with metformin. And this is really, really interesting, showing again the need of E. coli for effects of metformin on host physiology. But at that point, we know that high-fat diet has changes the microbiome, increases the abundance of E. coli with metformin, but we don't know exactly the mechanistic, what is the mechanism behind this. So we turned back to doing experiments with C. elegans, and what we did here was to mimic that a little bit. So this is a complex scenario with a lot of data points here. So the first thing we could do was test several concentrations of metformin. What you see on the y-axis is mean lifespan. Normally, people just would do one thing, which is the zero control, no drug, and one concentration, so 50 millimolar. We tested everything, so we could have these beautiful curves. And what we changed then was the nutritional content on which these worms are growing. So we took different types of media, either a peptone that has an animal origin, a plant origin, a complex peptone, or even a rich glucose protein media called MRS. And you can see very nicely that the response and longevity on the host is completely dependent on the nutritional that these worms are growing. And quite remarkably too, that these effects were completely obliterated if we use, instead of using a bacteria that responds to metformin, we used a bacteria that does not respond to metformin. So this beautiful bell-shaped curve that we had previously described, if you put it on a resistant strain, they just simply, there's no positive effect. But of course, these are complex media, and we needed to move into something a bit more mechanistic. And the way we did that was to, around the 2018, there was this idea of creating a lot of foods, and this still is ongoing, of creating macromolecular foods that by knowing their composition, we could put these things together and try to recreate new foods that don't have, for example, are not from direct animal origin, right? But I thought in terms of science, we should be going the other way around, which is to really try to understand how each one of these different things really influences the relationship between microbes and host, and especially in the context of drugs. But diet is incredibly complex, and we've talked about a lot of the microbiome, the genome, and these are very complex things. But similarly, foods are very complex compounds, and some of them can have. There's been described about 150,000 at least different molecules that we ingest on a daily basis, and yet we only know really the effects of about 100, which are characterized by the FDA. So there's a lot of work to be done here to really understand how different nutrients have effects on host physiology, and how the combination of different nutrients can have effects on host physiology. So we try to do that, and again, we use the worm to achieve that. We combined this technique that was used previously in microbiology called biolog, and each one of these plates, 96 salt plates, contains an individual nutrient. And we try to understand how these different nutrients were affecting the effect of the drug, both in growth, whether they were synergizing or antagonizing the effect of metformin in growth. Metformin is known to impair bacterial growth, but we're trying to find metabolites that can alter that. But at the same time, we developed a four-way screen where we were trying to understand whether if you use the worm now as a readout of the effects of the drugs in bacteria in the context of nutrients, where we're recapitulating what we had just seen without the worm. So to do that, we created a transgenic worm line called ACS2. This is a gene involved in the activation of lipids to the mitochondria for lipid oxidation. And when you put bacteria that is sensitive to metformin and combine it with the drug, these increase the expression of this transgene, and the worms go bright green. And then we were trying to understand exactly how the nutrients make this, the expression of this transgene go higher or lower, and this is what we're trying to understand. And we measured about 5,000 phenotypes. So this is incredibly complex data. I'm not actually going to go over it, but what we just the bottom line is we could really separate different nutrients in their effects on host physiology and bacterial physiology. We can cluster now these nutrients in a similar way as we had done into the 2017 paper. Instead of putting it in the genes into pathways that has been done before, that was easy to do. But here we were actually thinking along the same lines, but no one has done that before. So we start putting, instead of the genes that are usually in the pathways, we start placing the metabolites that are often in those pathways to do enrichment analysis. And we can do two types of enrichment analysis, both the type of metabolite class, so whether they are carbohydrates, carboxylates, peptides, etc. But we can also start looking at the pathways that are being involved for each one of these different nutrients. And what you can see is that some nutrients can have an antagonistic effect on E. coli and also in C. elegans, and other nutrients will have a synergistic effect in E. coli, potentially also in C. elegans or not. But what we did find that was very striking is that some nutrients that belong to some classes antagonize both E. coli and C. elegans, while others could antagonize singly just E. coli. So how can the bacteria make a distinction between these different signals to regulate the host? At that point, it was very clear that this was the key question. And when we looked at a functional enrichment in terms of pathways, we found a metabolic pathway, galactose metabolism, which antagonized the effect of in E. coli and both C. elegans, and a signaling pathway, so the phosphotransferase system pathway. And here we have basically a metabolite pathway or a metabolism pathway and a signaling pathway. So how can the two work together? And this was really something we wanted to know. So in order to know that, we performed proteomics in bacteria treated with metformin. And what we did was just simple expression changes in a treatment condition versus a control condition. And we found then several genes being changed. But what we did with that information was, again, more enrichment in terms of function. We found glycolysis to be involved, TCA cycle to be involved, and arginine degradation to be involved. And we took those genes and asked, because in E. coli it's known what are the transcription factors that regulate the expression of those genes, we can take a layer of information computationally about what are the transcription factors that are involved in those expression changes. And we found 4 major transcription factors: CRP, CRA, ARGR, and NTCR. And we can also do a functional understanding what is the role of these transcription factors in the regulation of these pathways. So with the 4-way screen and now the proteomics, we found basically an entire signaling pathway in bacteria that is activated by metformin and requires this transcription factor to regulate the effects on host physiology. So we tested that. So we took C. elegans with growing in bacterial control conditions. This is a longevity curve. You can see the worms over time, they die. But when you treat it with metformin, you see this extension about 33%. But now if the worms are grown in a bacteria that doesn't have the transcription factor in E. coli to mediate these effects, you can see that metformin actually doesn't have a pro-longevity effect anymore. So at that point, we were very curious, and we wanted to do almost perform epistasis analysis that is often done in C. elegans genes, where you try to understand a signaling pathway like the insulin signaling, where you have a deletion of the receptor in the membrane like DAF2, and then you show that FOXO, the transcription factor, is required for that pathway. So we wanted to show the same thing, but now playing with bacterial signaling. And we did this by overexpressing the transcription factor. And you can see that when you overexpress it, you can make worms live longer. So analogously to what we saw with metformin, when you activate the CRP pathway. But if you remove the SIA-A gene, which is important to produce the cofactor for that transcription factor, the effect was completely gone. So this really worked beautifully and really showed that the activation of the signaling pathway in bacteria, even either by metformin or genetically, was sufficient to increase longevity in C. elegans. But of course, how is the bacteria now communicating with the host to produce these effects? We did a lot of metabolomics analysis, and what you can see, and this I'm just going to focus here. So we treated, these are treated cells versus non-treated cells of control bacteria. We see a lot of changes in metabolites that are highly abundant versus others that are underabundant. But if you don't have CRP, basically the bacteria are completely blind to the effect of metformin, and there's barely any significant changes in metabolites. If you overexpress CRP alone, in the same way that I showed in the previous slide, at different concentrations, we can now start seeing a signature that is actually very similar to the effect of metformin. When we did further analysis of what metabolites were changing, it could be interesting, and this was a very humbling discovery. We found metabolites that had been described almost 100 years ago, where people, before the discovery of metformin, were giving to patients to treat what was known at the time like type 2 diabetes. So, and we focused on a metabolite called agmatine. So, and at that point, we collaborated with a group in Germany to really try to understand how and whether agmatine can be produced in human cohorts. And for that, we used a computational approach that by doing metabolic modeling of the pathways that are known for each of the different species present in the human microbiome, and knowing the abundance of these different species by 16S, and having information about different diets from these patients, we can predict whether a specific metabolite is being produced by the community. So, and we can ask other questions too. What are the most important microbes in the human microbiome that can produce this agmatine? And we found in several cohorts from different countries that Escherichia coli was actually the most important producer of this molecule that we had previously shown to increase longevity in the presence of metformin. We even checked whether there was a significant difference in agmatine production between healthy patients and type 2 diabetic patients with and without metformin, and we see a small but significant increase in agmatine production in type 2 diabetic patients with metformin. In a longitudinal cohort from Spain, you can see that on placebo, there's no changes in agmatine production without any drug. The moment you put them on the treatment, you see a significant increase in agmatine production, which mimics patients that are being taken metformin from the very beginning, which increases over time. And we can ask other questions. What are the other molecules that are produced by this microbiome together with agmatine? And you can see the production of different gases, CO2, hydrogen sulfate, but also some short-chain fatty acids, which can explain some of the positive effects of taking metformin, but also some of the side effects that are accompanied by taking this drug. So is agmatine extending longevity on its own? Does it have that property? And we did this both in C. elegans and in flies. And we can see that if you supplement agmatine in worms that are colonized with bugs that can't digest agmatine, we can robustly extend longevity. And the same effect was observed with a fly model. So these force waste screens reveal really new mechanisms that could never been revealed in any other way. These mechanisms were then supported by different techniques. We found, and I think this is really exciting, that metformin can activate signaling pathways in a very analogous way that we see in host cells. So again, the host cells and the bacterial cells seem to mimic each other and sometimes communicate. And this is a great example of that. And also we could see that a lot of these effects were conserved in different organisms, eventually explaining the human effects. Just quickly now, these are four or five slides on our last study. So we'll start studying, we had a focus on antipsychotic drugs, but we started this study by looking at different drugs that are normally taken for different types of diseases. What you see here is a PCA plot, and the way these are done is each molecule has a molecular fingerprint, and we can transform that into a vector that we can then perform a PCA on. And what you see here is basically to show that we picked drugs that were representing quite well the chemical space of all the different FDA-approved drugs. So this is not a biased study towards a specific type of molecule or a specific type of functional group within these molecules. And we tested these 15 selected drugs with 25 different strains from the human microbiota, and we looked at two different things. So on the right-hand side, in terms of growth, whether it impairs growth, here is shown in red, or whether it actually improves growth, so not just reducing, but also improving in green. And as you can see, a lot of these drugs tend to have either an inhibitory effect rather than a promoting growth effect, although we see a few that having do that. But what we did discover, and on the left side is actually one known mechanism, which is a biotransformation, which I mentioned about 5FU, but also what we discovered here was a new mechanism called bioaccumulation. In particular, diloxetine, which is an antipsychotic drug, with many different bugs was basically being bioaccumulated. Although this mechanism is not just for duloxetine, we can see it for montelukast or isoglycosine for specific bugs. But duloxetine, a large proportion of bugs did have this ability. So we developed some mass spec assays to really show that point. What you see here is just this spectra of bacteria alone, bacteria plus duloxetine as the spectra. If you give just the duloxetine without, you see even bigger peaks here. And when if you do you can see that when you subtract the signal from this one or this one to this one, you see a very clear signal there showing that indeed microbes are accumulating duloxetine. We tested this with different bugs, and you can see indeed that if there's no bacteria, the abundance is 100%. The moment you treat it with different microbes, you can see a decrease in the abundance of duloxetine, which shows really this bioaccumulation. But what are the importance of this bioaccumulation? Well, one of the things that in order to understand that, there's several techniques that were done. So click chemistry, pull downs, and thermoproteomics, and also metabolomics. And all of them seem to be pointing out of a change in enzymes that were involved in purine and pyrimidine metabolism, but also in the metabolites that are involved in these pathways. We went further to understand whether this was important in any way. And in a way to do that, we perform an analysis of the metabolites that are secreted by microbes, and we can see that there's a shift. So each one of the shapes is a different microbe, and in gray we have the control, and in red the treated, and you can see a shift into of the exometabolome of all these different microbes when they are treated with duloxetine. So the duloxetine gets in, gets trapped, interacts with some enzymes as shown here by click chemistry and thermoproteomics. So it's changing the ability of some of these enzymes to function. This changes the abundance of metabolites, and this is recapitulated into a change in the exometabolome. You can see here very nicely with just for E. coli that increasing concentrations of the drug change the exometabolome in a dose-dependent manner. So why does this matter? Well, if you have over time, you have an inoculum with different, a population, a small community of 5 bugs, you can see that they establish an equilibrium eventually, which is mostly made by B theta and salivarius. But if you increase now, if you actually add duloxetine from the beginning, you can actually see that it completely changes the community structure of the microbiota. And finally, we tested whether really this bioaccumulation, this hoarding of the drug was changing the effect of the drug on the host. For that, we use C. elegans. You can see that increasing concentrations of duloxetine, because this antipsychotic drug affects the movement of C. elegans. And you can see that in a dose-dependent manner, we can see very clearly that if you give duloxetine at 50 or 500 micromolar, it impairs movement. But if you put a bacteria that accumulates and reduces its abundance for the host, we don't see this change in movement. And this can be done with 2 strains, one that bioaccumulates and one that doesn't. And this is very nicely recapitulated. So the summary of this study is really we've been able to find new drug-microbe interactions, in particular bioaccumulation, which had not been described for therapeutic drugs in the microbiome, that this changes the exometabolome, and the consequences of that are that the microbiota structure changes, and this is working through a cross-feeding interactions. And we finally show, and this is potentially important for host physiology and for humans, is that it reduces drug efficacy. So I just want to thank my team and my collaborators, of course, without which this would not be possible, and the several funders that over the years have supported our work. And here our Twitter account, if you want to follow us. We're not incredibly active, but it's something to work on, especially as we saw before. Communication with the public is an important function for scientists, and our websites, if you want to keep in touch or get in touch with us, are here on my emails. So thank you very much, and I'll take any questions if you have any.
Thank you, Professor Cabrero, for such a great talk. We have time for some questions from the audience. The first one is from Professor Modena. Do you think it would be possible to use the system to investigate the influence of bacteriophages and other viruses on the metabolism of drugs by microbiota-dependent manner?
That's an excellent question. It's funny. Sometimes you have to make decisions and choices, right? And this was something I wanted to do literally when I started my lab. There was a big thing at the time about bacteriophages, and I wanted to implement that. And we never got to doing it. I think it is possible. It would require a bit of dedication to make it work, but I think it's definitely possible. And I think it's ripe to be done and to make some critical discoveries using it. So it's a really, really great question, and I think it can be done. Yeah.
Okay, we have one more from Ricardo. When do you think these treatments using engineered bacteria could be made available?
It's a very good point. I don't know. These are GMOs, right? So are we there to accept genetically modified organisms and take them? The legislation is not there. I think there will be a push more and more. I think we need more evidence to support that these are, that they do what they says. We can show that in animal models, sure, but humans tend to be quite more complicated. So, and also that they are safe, right? Synthetic biology has moved a lot, and we can include really safe systems into them, like fail-safes inbuilt into the microbes that we can, by adding a specific compound that is not harmful to the host, we can destroy them and eradicate them. So there's ways, and I think we're all doing steps in the right direction to achieve that, but also our perception about GMOs will have to dramatically change for these to be widely accepted. So yeah, it's a very good point.
Okay. We have a question from Ciro. Is it possible to establish a correlation between a specific diet, like vegan, vegetarian, with the metabolism of drugs?
Yeah, it's a very good point. I have to say it is. Correlations, absolutely. Causation, it's where the work is really going into, right? We're really trying to work out. The problem, as I try to say within the talk, is even a diet like vegetarian is complex. There's more than one thing in there. So what in that complex food has a better role in antagonizing or synergizing or transforming it? We don't know, and this is really the big challenge. But already we can see from human studies that big bulks, like between vegetarian or carnivorous diets, we can see very big differences already in drug metabolism by the microbiome. And this can be recapitulated by either in vitro work or by transposing this into animal work, actually.
Mm-hmm. Okay, we have one more question from Modena. Do you think that acetate and other short-chain fatty acids produced by the microbiome during metformin treatment could contribute to this increased longevity?
I think it's very likely. So I didn't show in the work in the study here, but in the published paper we went on to understand on the host side. So here I focused mostly on the bacterial side, but on the host side we looked at what kind of mechanism was being activated by agmatine, but also by the metformin, and we see it has to do with lipid metabolism. We saw that the homologue of PPAR alpha in humans, or NHR49 in C. elegans, was required for almost the totality of these effects. And analogous to the human context, short-chain fatty acids are important to act also on the PPAR alpha or the PPAR or hormone receptor. So it is very likely that this is a combined response from agmatine plus short-chain fatty acids, and the total effect of these working through the PPAR pathway would have these longevity effects. We didn't test that in C. elegans, and I'm not even sure that actually people have checked with short-chain fatty acids in longevity in worms. I need to check that. But in the context of humans, I actually believe it's the combined action of these different things. It's a good point, a very good question.
That was our last question.
Brilliant. Well, thank you very much. This was a pleasure.
Thank you, Professor Cabrero. I hope to see you soon.
See you soon. Goodbye.
Bye-bye. Seu microfone está mutado, Ricardo.
Okay, now for our next talk, I'm very pleased to announce our next speaker, Professor Ivan Costa. Professor Ivan works at the Institute of Computational Genomics at the University Hospital RWTH Aachen in Germany. He holds a bachelor's degree and a master's in computer science from the Federal University of Pernambuco, and a PhD in bioinformatics from the Max Planck Institute of Molecular Cell Biology and Genetics in Berlin. Professor Ivan has wide experience in computer science, focusing on computational biology and machine learning. His research is currently focused on computational methods for the analysis of epigenetic and regulatory mechanisms of dynamic processes, such as cell differentiation, aging, and the onset of diseases. Professor Ivan, thank you very much for joining us today. You can start your talk.
Okay, yeah, thank you. It's a great pleasure to be, talking, I think, for a Brazilian audience after actually maybe potentially more than 10 years, even if it's still virtual. So today I'm going to talk about, let's say, a few computational methods, in particular exploring single-cell methods, and how we are using these together with collaborators here in the University Hospital to try to understand mechanisms driving diseases. And I mean, maybe with this title, the slide is actually omnicellular, and this goes back to Virchow, who was actually one of the founders of pathology. And what Virchow comprehended at his time was that he started using a very modern instrument here, let's say a Zeiss microscope, to look at cells of patients and trying to understand then how diseases are upcoming. And he came with this phrase, which means simply all cells come from another cell. And this is shown actually in this system where, which is one of my favorite systems, hematopoiesis. And this is a system that shows how a single hematopoietic stem cell can actually differentiate into further cells and giving rise to all blood cells or immune cells in our system. And when we are looking at this system, we want to understand what are the molecular players, right, making these decisions. We know, for example, transcription factors are proteins which are very important on those processes. But we also know that there is something happening behind, which we call cell memory, so that somehow a cell, after being, let's say, pushed toward these decisions, it remembers that it's a T cell and should only, for example, express T cell related genes. And so we would like to have now methods or computational methods to try to understand how cell differentiation is working, but also if can be, in the case of diseases, how this misspecification can happen. And coming back to the cellular memory, what we know is that actually the chromatin is one very important mechanism in eukaryotic cells. And in a very brief way, what chromatin is about, let's say, mostly that the DNA is really not, let's say, just a naked molecule, but actually is wrapped around these proteins, in particular these histones. And there are, let's say, two major types of chromatin. One is the condensed, where actually the DNA is wrapped around the histones, but further condensed. And here, if a gene is in this area, it's very difficult to express. While if it is in the decondensed, it's, let's say, this chromatin is more accessible, so particular proteins like transcription factors can access particular regions, remove histones, and, for example, start transcription of this gene. And the chromatin structure is very important now to, for example, to make it that the monocyte doesn't express, for example, T cell related genes. So if we want to understand this, then we should look at chromatin, and this is something that, for example, we are doing a lot in the lab to, for example, use protocols like ATAC-seq to understand where chromatin is open. And usually chromatin is open, just coming back to the slide, in regions where we have transcription factor binders, right? So, for example, in a promoter protein, but also in these distal regions, which enhancers. And in these regions, actually, the size of this naked DNA is larger, and one can use things like these molecular scissors, for example, this TN5, which actually breaks and put adapters in the DNA whenever the DNA is accessible. And you can use this as a fragmentation. This protocol is, of course, a little bit more complicated, but making it simpler, well, you can fragment your chromatin, and you can isolate, for example, only the beginning or these small fragments. And if you put this now in a sequencer, you have the so-called target DNA sequencing. So you should see a rich enrichment of reads around regions where DNA was most accessible. And another thing is that actually, if you have a TF binding here, it should actually protect this in this case, the TNF5 to cleave here. And this will make it that we will have a footprint. Of course, you have, when you're generating the signal, you have to do now special counting, so you only count the beginning of the reads. But if you do this, you should see these, let's say, small values around regions with high number of reads, and these are indicative of transcription factor binding at a base pair resolution. And if you can find these, you can now go back, for example, to the DNA sequence and try to figure out which factors were these. And out of this, now we have one single protocol that can help us to understand the regulation in one particular cell. And this is something that we have been working actually for almost 10 years now, and where we were using computational methods, which are based on hidden Markov models, which if you give a signal like this, which is, let's say, the ATAC-seq, would look for patterns, in this case, let's say small values in regions we have large number of reads. And this is actually prediction. So these blue regions that you see here are the footprints, the regions that we predict that there was a binding site because it was, let's say, protecting the DNA to be cleavage in this area, which looks like an enhancer, and here, for example, is a promoter of this gene. And now we can go back to motifs. So we have databases where we have motifs of up to 500 transcription factors, and we can just scan this in this region. And whenever we have things like this, for example, here you have a binding site of this protein, which is CTCF, which is really overlapping well. Then we believe this is a TF binding site that is active in this cell. There is also other CTCF binding motifs which are not really associated to open chromatin. It might be this important in another cell, but it wasn't predicted to be important in this cell. So out of this, we can now infer cell-specific binding site, even though computationally, but this is much better than before, where because at least now with the footprints, we can really know where to look for. But we also can do other things. For example, what we have noticed is that usually this is only one region, but we have dozens or thousands of these binding sites. We can do this kind of average profiles, which is just like, say, the accessibility around all the 10,000s of CTCF binding sites that we are predicted by, which are supported by a footprint. And we can measure the TF activity, which is kind of the difference between the value, right, the footprint, and the open chromatin around it. And what we have noticed is the highest this is, the more likely that this factor is doing something in that particular cell. I will come back to this one. So, because of course, let's see, if you remember my first slide, we wanted to understand things like this, a cell differentiation process, right? And these are cells, again, blood cells or immunological cells, and some of the cells are similar to the ones I showed before. And we have, together with the cell biology, we have done different chromatin and gene regulation and gene expression experiments here. But here, for example, I'm only showing data on ATAC-seq, where we measure now ATAC-seq in all these 4 cell types. And we wanted to particularly understand how is this decision here? Why do these cells either go to this type of dendritic cell or to this other type of immunological dendritic cell. And the way we can do this is like this. So we can, here we are only comparing pairs of cells. So let's say these two cell types. We measure the footprints of the both cells, we combine them, and we run all factors that we have a motive, which is more than 500. And we measure the activity either in one cell or in the other. And then we measure the difference in activity. And then we can get this kind of plot. So if a positive means it was high in PDC, negative means it's high in CDC. And for example, take here TCF4, you see that the footprint is larger, indicating what's more accessible in the PDCs. This is why we have a positive difference. In the other side, we should see the opposite, right? So a higher red signal, which indicates that this factor here was more specific to CDC cells. And indeed, look in the literature, and we see that, for example, all these factors which are indicated here in green were known to be associated already to this differentiation, but we could predict some new ones, like this DCBTB18, which actually was confirmed 2 years ago after our publication also to be important in humans. I mean, all this data here is in mouse. So we can now predict, of course, novel, known things, but also novel things. And the other important point is that we can really go back now to the individual sites, right? So we look here at peaks of ataxic. Here are CDC1 specific, so only for this particular cell types here. We look at genes that we knew to be important in these cells, and we could find, for example, several enhancer-like peaks, which have footprints, which, for example, have a motif of BATTF3, which was this factor here that we predicted. And we have ChIP-seq data for this factor, so we could, and this is, let's say, the best experiment that you can do to confirm, and we could see that indeed the binding site, that we had BATTF ChIP-seq signal in these two predicted binding sites. This also as a display that we can really go now back to the individual, let's say, enhancer level. So, of course, all this data here was still only, let's say, in the immunological and just trying to understand, let's say, cells in a normal state, right? But we would like to maybe further go to trying to understand diseases. And one thing that we're exploring now is that, for example, there are new technologies, right? So, for example, Vico was using a microscope, and this is how he was able to start characterizing cells. But now we have things like single-cell sequencing, which are now allow us to do either RNA sequencing, but also taxic sequencing in the single-cell level. And we can now try to really see how these cells are differentiating, right? And this is really one of the first single-cell taxic data which was around, which is explaining exactly this system, the hematopoiesis. Not sure, maybe I don't need to explain to all of you. I'm also not an expert in the biological part, but the idea of this, in this case for taxic, is that you do your fragmentation of a chromatin before you put it in the single cell machine. You have your cell suspension, which is already now, let's say, fragmented, or your chromatin is already broken. And then you put in these machines, which actually have the droplet in a cell. And this machine makes in a way that you only should have one of these, one cell per droplet, and also one of these bubbles here, which is actually a chain, which will have now a particular barcode, which is different from each other chain. And then in this machine, we can do the library preparation so that these barcodes, which are different for each of the genes, are incorporated in your DNA sequence. So now we have a combination of a read which was associated to open chromatin, but also the barcodes. And with these, we can now try to build up something like a matrix, for example, which is now where in your cells you have open chromatin. So the problem here for open chromatin is that usually you don't know where to look at in the beginning, right? So for example, if you do single cell RNA sequencing, usually you know the genes for the human genome. Here, we don't know. What you could do is you can ignore the cell information, look at the peaks, which are, let's say, the regions which ignore that you have single cells. These are the regions which are potentially, which should be open now in the individual cells. And then using these as your feature space, and then you can just now count how many reads you have per peak. And of course, because you only have to copies of a DNA. There is a theoretical maximum of 2. These peaks can be a little bit larger, so sometimes you have 10 reads, but overall this matrix is extremely sparse and the values are very small. And once you have this, then you can try to do things like clustering. And once you cluster, then you can do what we call pseudo-bubble attack. So you can go back, just combine the cells from a particular cluster, and then do all the analysis which I have shown before, which were based on bulk attack sequencing. You can now look at the open chromatin in the gene, but also you can look at this footprint profiles, which indicate activity of a particular factor, but now in these different subcell types. So yeah, but the problem that we have seen when we started looking at single cell data is that this data is extremely sparse, meaning that first, the dimension here is huge. So if you have a very complex tissue, you will easily come up with more than 100,000 of peaks. And this is a very bad, I mean, this makes it a very difficult problem in the point of view of statistics. And the other problem is this, that most of the entries here are going to be 0. Potentially because most of this, both partially because most of these regions are not really open across all the cells, but also because you have a loss of DNA material. So maybe only 30% of the DNA in the end really goes up to sequencing. And this makes it maybe at least, you should have at least double or 3 times the signal that you see here, but you cannot see it because losses in this whole process of the single cell isolation and then library preparation and the sequencing part. And to solve this then, and this is what we went to try to find ways to solve this in a computational approach, and also making a very, let's say, complicated story short. One has to get to this count matrix. There are transformations that we do, which are based on TF, IDF. It's a transformation that's usually used for document or text mining, which also tell us, for example, which peaks are more relevant. And usually peaks which are relevant are not the ones which are shared across many cells, but that the ones were actually rare peaks, so ones that are not only in a few cells. And we do this in a, let's say, in an unsupervised way, without really knowing which cells are there. And then we use a non-negative matrix visualization method that tries to input this data by reducing this matrix in a smaller space. And once we do this, then we believe that we have, let's say, a denoised matrix or an inputted matrix that you should have more noise than the original one. And you can now just give this matrix as input of any kind of further downstream method that you will use in single cell data, as clustering and visualization, but there's also motive or TF activity analysis. Okay, so how does this look in reality? So for example, here we got this data, which I think I showed before, which was the first data in single cell attack over hematopoiesis. And these are different tools which were out there. So PCA is, for example, the most standard dimension reduction you can do. If you do this, and we have labels here because the cells were actually initially first isolated in the sequence, so we really knew their identity. And PCA doesn't really work, or if you just take the raw data, also doesn't really separate well. But there are different methods that are working well. For example, Cystopic is a competitor. And you can see here that we get kind of the trajectory. So here's the HSCs, which is the stem cell. You have CMP, which is kind of this way. And you can see now, for example, the map cell. So you can really find trajectories here. And SPC Open was actually having the more clear trajectories, which we could also validate by, for example, doing clustering in these dimension reductions. And we could see that the clustering accuracy was higher for SC Open comparing to the other methods. We have a large amount of benchmarking with a lot of the single cell attack to show that indeed doing denoising with SC Open was helpful. But I will maybe skip the more technical parts and focus on something that, let's say, more of a biological application, right? As I say, we went now to try to use this method to understand diseases, and together with medical specialists here, which are interested, for example, in kidney diseases, we wanted to investigate what happens under kidney disease. And one aspect they are particularly interested is the fact that, let's say, upon diseases on the kidney, over time, patients tend to have what we call fibrosis. And let's say the kidney looks like this, the structure. You have, of course, a capillary, so this is trying to somehow filter your blood. And the tubulars, these epithelial cells where your urine will go through, and you try to, let's say, clean the toxins which are in your blood. But whenever you have this kidney disease, you have one of the things you can have with fibrosis is has to do with that fibroblasts, which are cells which are usually filling up spaces in organs. They can get hard or start to scar. So just like when you get hurt, you get a scar. But you get a scar inside the organ, and this makes it that it's going to be stiffened, and then of course the physiology of the organ will not work so well, and this usually leads to kidney failure. And these colleagues want to understand different things. Let's say which cells are really starting to become these fibrosis-causing cells, which are called myofibroblasts, and also what events drive this, right? And here we wanted to look at toxic because then we can understand the regulation. And we have already some understanding of how this is working, and usually it's when upon some sort of injury, which is coming from your disease, these tubular cells here, they will de-differentiate, will release in particular TGF signaling. This can activate, for example, immune cells, but also goes to the fibroblast cells, and they will then, for example, start differentiating these myofibroblasts and matrix producing cells. And we wanted to understand and have a very more clear picture on how this is going in single cell level, which wasn't really done, in particular using a toxic. And to do this, these colleagues use a mouse model where you block one of your kidneys. And this was done before blocking or without blocking, but also 2 or 10 days after the blocking. And then for each of these 3 days, we measure then single cell toxic data. And the nice thing is that the kidney has several different subtypes of cells, and we could find, I think, almost 14 subtypes of cells, and including major cells like the endothelial cells, proximal tubular cells, which are these epithelial cells making these tubules, and but also, for example, rare cells like these podocytes, which make this more fine structure here, which is called glomeruli. Yes, and of course we have time courses now. The first thing we asked was, for example, is there a change in the cells in population? And yes, we saw, for example, that immune cells, like say the macrophages and lymphoids, they were all increasing over time. We saw, for example, loss of endothelial cells and also loss of these proximal tubular cells, with exception of one population here, which had markers associated to injured proximal tubular cells. And these were all, let's say, hallmarks of things that people know. When you have kidney fibrosis, you should expect all these changes. And it was good to see that we are kind of seeing them back. Of course, we wanted to understand here now gene regulation. So what we went doing was for all the clusters now, we measure this TF activity, just like what I have shown before, but now using the single cell data. And here is a few selected cell types, which were of interest to us. And what you can see here, for example, there is a large group P of TFs, which have high activity. So red will mean these TFs are important in these particular cells. And what you can see, for example, that upon injury of PTs, you lose kind of several TFs, which are important to the normal cells. And this is going into this understanding that these cells get de-differentiated. So they kind of lose their identity. And for example, if we look at the footprint level, we also see this, for example, that these are 2 hallmark factors associated to proximal tubular cells. You see that they are quite high in PT cells, but they really decrease in these injured PT cells, and there was almost no activity in other cell types. What was quite interesting here was that there were actually a group of factors which were kind of shared between the injured PTs and the fibroblasts. Again, we want to understand fibrosis, and we wanted to look at this. And actually, when we look at these factors, we find things like SMAD and BATF. And SMAD actually is a TF which is downstream of TGF beta signaling, which was one of these pathways I have shown in the chart a few slides ago. And what we see here is that the signal is pretty high in comparing to the other cells and very specific to this injury, showing that these cells are really now driving the signaling, but also was increased next in the fibroblasts, indicating that this inflammatory kind of signaling is potentially shared between those. The other thing is that we can now look at one cell over time, right? And the interesting thing here is that, for example, what we could see is that in the early part of the injury, this TGF-beta or SMAD TF-activity was already quite high. While, for example, this kind of the loss of the identity was a little bit more gradual over time, indicating, of course, that some of the responses, in particular the inflammatory ones, are going to be quicker than, for example, this differentiated one. So then, as I understand, coming back again, we wanted to really look at fibrosis. So we try to zoom in these cells to understand, okay, is there anything more clearly if we try to subcluster them? And what we did is we did a different analysis here. So this is a diffusion map. So so what we do is only look in the fibroblast cells. We try to see if there is any kind of differentiation trajectory. And we could see something interesting, so that there was the cells which we could say this is, and they have also expression markers which are indicative of progenitor fibroblasts, and here there were the myofibroblasts, and these are the fibrosis causing cells. So we expect that this should then indicate this trajectory of how some of these fibroblasts are now becoming these fibrosis forming ones. There were also something interesting here. These are parasites, also a subtype of fibroblasts which are usually around endothelial cells. We also saw that they also come into these myofibroblasts, but in less amount. And because the data was sparse, it was difficult to do this trajectory, so we don't really show this. But when we also compare over time, we see also that these fibroblasts, so over time is here, that there was most of the cells here in the end were associated to the day 10, indicating that they were accumulating over time. And the nice thing about this pseudo-time, and now we can look at things like TF activity, and one thing that one also can do in ataxic data is we can look at also how open the chromatin is of a gene, and this is usually proportional to the expression of this gene, and this is what we call TF activity, gene activity, sorry, right? So the TF activity is associated to the footprint profiles, and gene activity is how open the chromatin is around one particular gene, which can be a TF. And here we look at these genes, so we order these genes by this pseudotype, like in this order, and we were particularly interested to know which TFs were having high activity and high expression towards the end. And we find, for example, TWIS2, which is a no EMT factor, but we also find, for example, RUNX1, which was a quite interesting gene because it has been shown already to be associated with fibrosis in other tissues, but never in the kidney before. And RUNX1 actually is a factor which is very well known to be associated with blood cells. And also just to highlight the importance of doing single cell data, if I go one slide back, you see the RUNX1 was in this heat map. It was actually very high in the immune cells as expected, but was already elevated in the fibroblast. And indicated that actually if we comparatively, RUNX1 still have high activity in lymphoid macrophages, but inside the fibroblast, at least in the subpopulations, there was an increase. And this is the power of the single cell data now, because we can really zoom in populations. So interesting to find the difference only between this subpopulation. Okay, so, and of course we wanted to validate this, and these colleagues did different validations. Here's, for example, staining on these mouse models, and we can see that so BDGF beta is staining this fibroblasts. RUNX1 is a marker potentially also of the immune cells. And we have expression already RUNX1 in the control condition, but when we go to this kidney injury, we see that we have much more expression of RUNX1 and it starts to co-localize with the fibroblasts. And if we look at this fluorescence intensity with co-localization, the fibroblasts, it's increased upon the kidney injury, showing that now we could really see that RUNX1 is important in immune cells, but also in the fibroblasts. We also have done all the experiments, for example, also knockout on a fibroblast cell in cell line in humans, also to show that this was also relevant in human cells. And also maybe going back also to computational aspects is, and maybe you have noticed that another thing that you can do with single cell data, single cell toxic data, is try to link now particular genes to peaks, right? Or in which might be distal, for example, 250 kb. And if you think about this chart, which I was showing in the beginning, it has to do about these enhancers, right? Enhancers are regions which are potentially far away from the promoter of the gene, but because the DNA can fold, and they will still be important for the regulation of that particular gene. And it has been shown before that actually, if you now have single cell data, what you can do is you can measure the correlation between the open chromatin values of your promoter versus enhancers. And enhancers that have high correlation are usually collocated. And they have been, for example, validated by looking at chromatin conformation sequencing protocols, which really shows how this fold is working. Here we don't have chromatin conformation, but we use this only for the prediction. But one cool thing, for example, that we could show the power of, let's say, doing the imputation. So if we, for example, these are all, let's say, this is TGF-beta receptor, right, which is of course important to be regulated if a cell should be sensing TGF-beta signaling. And you see that this gene has increased in accessibility over time. So this is pseudo time. So this is a differentiation for fibroblasts until myofibroblasts. And we see several peaks which are correlated. So they're usually merged with an arch here. And some of those also have a RUNX1 and footprint supported TF binding. Also the promoter, but also, for example, this one here, or this one here. And here, for example, this P4 is quite clear. You see that it's really opening, the chromatin is really increasing over time. And also the other nice thing is that if we look at this single cell values versus the gene activity, we see that we see a high correlation. So this correlation from this value between the one is 0.4, but without the imputation, without using SC open, there was no correlation. This is actually in this kind of analysis where the imputation will have, let's say, a more synergistic advance, because you input in both this peak and this peak. Imputation seems to be really having even a much stronger effect than, for example, by the clustering problem. So now we can really say, or we can start doing these predictions, right? So that RUNX1 is potentially binding here in this peak, and this potentially now, and this is a potential enhancer which is associated to the regulation of this gene, TGF-beta R1. We couldn't really do validation on the enhancer level. This is difficult. We need some CRISPR and some cell line that we didn't have for mouse data. But for example, what we did is we did this overexpression of RUNX1 in a human cell line. And we could see, for example, that whenever we do this, several of these genes that we have predicted as target, like TGF-beta receptor 1, have also increasing expression, right? Showing that at least that if you overexpress RUNX1, you also see a high expression of this receptor, which is fitting with this prediction. And we could see that this was overall also fitting, that we could really improve prediction of the target genes, at least supported by overexpression, even though overexpression might be also coming from indirect effects. Okay, so I hope you have all followed, right? So I have shown in this first part, let's say, actually almost 10 years of research in the lab, where we are starting to do these things like looking at toxic, doing footprint analysis combined with motif detection to predict cell-specific binding sites, going out to the single cell level, where we can first detect cells, which might be even very rare, like between a kidney, between a full organ, and then doing this more complex analysis. So one cool thing about single cells is that the sample size is large. So even in the fibroblasts, where we have something around 400 cells, we could do this kind of pseudo-time trajectories, and we could start trying to do inferences based on these temporal changes. And we could also, and this allow us, for example, to identify RUNX1 as a fibroblast driver, but also to identify some of the target genes, which is potentially regulated by RUNX1. And all of this based on computational methods, of course, using rich single cell data, but coming back to the lab to validate some of these predictions. Okay, so this was kind of the first part of the talk, and we have a second part, which is again on the single cell level, but we kind of looking at a bit different single cell problem. And this problem is, okay, how is it that the changes in communication of the cells, which is, for example, the signaling, is causing also diseases? And again, using single cell sequencing, but now actually going back to RNA sequencing, not necessarily a taxig. And I mean, I go back to another fibrotic disease, because among others, I mean, several of my colleagues here in the medicine, and I'm interested on fibrosis. And here is also another very important example. Again, I will try to show this. I'm not a clinician, but the idea is like this, that if you look at the bone marrow, there is in homeostasis, there is several signaling things happening, right? And you have these MSCs, or fibroblast cells, which are either attached to the bones, others are, for example, attached to the endothelial cells, which are, for example, parasites in the kidney. You have your hematopoietic stem cells, and of course, this is differentiating and giving rise to immune cells. And some of these immune cells, which some of them will leave the bone marrow, but some stay here, and they are also doing some signaling, and they are very important to doing this homeostasis, right? But whenever you have some hematopoietic diseases, and we are particularly interested in these MPNs with myeloid. So these are hematologic disease associated to myeloid cells. What you have is that some of these HSCs, they start to be clonal, right? So they acquire mutations, and this will disimbalance the differentiation process, right? So this process of cell differentiation is going to be biased. So either you're going to have too many of some cells or too few of some cells. And in the MPN, usually you have more of these megakaryocytes, which are not also too many, but also they misbehave. They become dysplastic. So they start different signaling, for example, TGF-beta, and when we have TGF-beta, then some of these fibroblast cells, they would start detaching, and then you're going to have bone fibrosis. And once you have fibrosis, so all your blood cells, they are usually born inside the bone. So if you have a fibrotic tissue there, you're not going to have space anymore to have cell differentiation, and then these are going to actually go to a leukemia, and of course then is leading to that. And the interesting, and here one question is that actually we know the MPNs, some of the MPN subtypes are chronic, but they can be treated. But at some point, once we have fibrosis, then this process is very difficult to revert, and then there is not many treatment options. Plus, it's very likely that the patient will have leukemia and die out of this. So it's will be important to know why, and it's only some patients that this MPN and these hematological chronic diseases will go to a more severe disease that will cause death. So, but to do this, we wanted to, in particular, look at how this cell communication is changing, right? And one way to do this is that we know that the cells are secreting particular proteins, for example, ligands, and they also will have receptors, and they are sensing these receptors. And whenever we have a cognate receptor, which is sensing a particular ligand, then we expect that there is a communication. And if you have some sort of time series, so if we can measure single cell data over time, we can see what are the changes, right? So not only that there are particular novel cell types, for example, the myofibroblasts or this plastic megakaryocytes, but also how the communication is changing. Yes, and in particular, we wanted to focus here in this problem, right? So can we infer now cellular communication or cellular crosstalk? Yes, and for this, we have done a deep characterization. So actually, we have mouse models where we could do single cell data, but was quite nice is that we have not really many cells, but this was, but we could recover a few hundreds of cells, which were associated to 5 different major populations in the bone marrow. And this was very difficult because, I mean, the biopsies, or usually you don't have a lot of material of this of the patients, but we were still able actually to find a few cells and to characterize these cells. And of course, we have this both in control and in disease, which we call here PMF, right, which is primary myelofibrosis. So one thing which we start to do was, for example, to see at the pathway level what's happening, right? So these are, for example, the 4 cell types that we have, and these are different pathways. And red will mean that there is increase of this pathway in disease. And we see, for example, MSCs here, and here is a bit confusing because the fibroblast cells, so I call here MSCs, which are, let's say, another type of mesenchymal stem cells, which are occurring in other tissues, but here then this would be, let's say, equivalent in a way to the fibroblasts. And this cells population here was doing associated to TGF-beta signaling. Also, we see a lot of JAK-STAT signaling, which is quite interesting also because in this MPN, actually mutations are usually associated to JAK-STAT signaling. And so in the end, this is actually this inflammatory cue, which is kind of starting the whole disease. And another thing that was quite interesting in this paper was also that we wanted to find also early prognostic markers, and we find, for example, alamines, which again, ligands, which are actually extracted by these cells, to be very highly expressed in all the cell types. And we could also use this to validate in patient plasma. And right now, our colleagues are also exploring this either as a programmatic marker, but also trying to treat these patients by, for example, blocking this ligand. But this is just a quick overview on several things that we have looked at this manuscript here. And this was just introduction to the system, because what I wanted to talk now is more about this. If you have now single cell data, you will now try to understand the molecular throw, right? So that some cells are doing communication by, for example, doing signaling via particular ligands, which are going to be now perceived by particular cell types. And actually, this idea is not really new, has been already there for a while now. And the idea is like this. So there are databases where you can now know cognate ligands and receptors. And of course, if you do single cell data, you will have a single cell matrix, which, for example, you can group a cell type, right? So you can cluster them first, and then you can have average expression. And then these methods were trying to do some statistics behind, which I will skip here, but whenever, for example, there is a cognate pairs of ligand receptors, for example, this to this one, and it was highly expressed in this cell and these cells, then we can draw a line from this one to this one, indicating that there was this cells was spilling this ligand, and it was sensed by this receptor. So we can now do an edge from this one to this one. And the same thing now we can do for all the other pairs, right? So for example, this other cell type here was also expressed a little bit less, but of this ligand, which is also sensed by the same cell here. And we can also somehow use the amount of expression as a weight, right? So of course, the higher the expression of this ligand here, then from this cell, then you should have a higher communication score than this one here. And these methods, or there are several methods that are doing this, and you can look at this review here, which is very comprehensive. Well, we try, but these methods are still, well, we saw what they were limited, among other things, because these methods, they were mostly only listing a huge list of potential interactions between cell types, ligands, and receptors, hundreds of them. And we would like to somehow first explore the fact that there is a network-like property when the cells are talking to each other. But also, since we're interested in disease, we like to compare, let's say, what happens, how is the cell communication in the normal condition in contrast to the disease. And this is what we went to do. So we have, let's say, assume you have a single cell data, you can group it on your normal and your disease. And then you do your ligand cell communication analysis using one of these conditions independently, using tools which I have shown in the slide before. But now, what we wanted to do is now to try to see if you can now compare these networks, because we have the same cells in two conditions, or mostly. And we can say, okay, because for example, it might be that this cell stops signaling here in the disease, and this cell starts signaling, but through other ligands. So what we tried to do was we started to now try to represent this data as networks, either cell-gene network, so which will be a cell and a ligand, a cell or receptor, but the cell will be combining, let's say, all potential ligands and receptors which are associated to one cell to the other, but also keeping the direction, right? Because we know a ligand goes to a receptor, so we can keep this in the network. And to do this either in the single cell level, but in particular to compare now to network, given that we have the same cells, genes in both of them. And another thing that we try to do is we try to use network properties to try to think about, okay, what are the important players in these networks, right? And we're going to give some very rough ideas how to do this, as some of these properties are actually very classical in using network-based models. And for example, one of them would be influence score. So if a cell is sending a lot of ligands, so it's possibly influencing a lot of the system, right? And so there is a lot of signal going out of the cell. And we have mediators, which are maybe cells which are sensing a lot of signal, but further sending signals. And we may have other cells which are listeners. So they're just listening to different, to having a lot of input from other cells, but not necessarily sending further signals. And we can also use other things, for example, importance score. Here, one uses a random walk. So you can start randomly in this network. And genes which are mostly visited, cells which are visited a lot, should be the ones which are more important. So now we can have all these different measures, and we can try to use this to uncover really either cells types or genes which are important in the cell communication. Okay, so now we'll go back to this single cell data. So we have these 5 different cell types, and this is one way that we can show now this network. So if we have an edge here, and the thickness of the edge will say how many interactions we predict, so how many ligand receptors. The darkness is about how many the expression of these ligand receptors, and the size of the node here is importance, right? And what we could see is that, for example, MSCs, which we know these stem cells, these fibroblast MSC cells are usually very important for homeostasis. They usually have very important score. But it was very difficult now to really compare both. So we came up with a way to try to compare both. So now we are looking at a relative scale. So our communication score now have 2 values, right? So orange will be increasing disease, blue is decrease. And what we see is most of the communication is lost, so that we are really losing this homeostasis. But we have increase, and increase was in particular the megakaryocytes. If you remember the chart, we have megakaryocytes are the cells which in these MPNs are really starting to drive signaling. And it drives a lot of signal to neural cells, which was for us a bit surprising. We don't really fully understand this, but also through MSC cells. And megakaryocytes were, from the importance, the most important node here. MSCs were actually decreased, but I guess because they lost a little bit of this homeostasis, but still they are receiving a lot of signal. So again, as I show here, this was fit into what is known in the disease, right? That this dysplasia of this megakaryocyte is the hallmark of these MPNs. And so it's not a surprise that most of the signal is really coming out of these cells. Okay, here I was looking at this higher level analysis, right? So cell to cells, but we have ligands and receptors, so we can really now zoom in. We try to look at these networks now, which is now this pairs of cells and genes. But when we look at this, it was a little bit like hairball, so it was really difficult to make sense of this. So we came up with a different way. So we do embedding. So we kind of do a PCA of this topological measure that we have shown before, right? So this influential mediator, listener page rank score. And then genes which are, or genes and cell parts which are sticking out of the center should be the ones who are changing. And because it's PCA, we can really go back to the features, and we can know, for example, here is influencer, but this is high influencer with increasing disease. So if I go in the other way around, it will be decreasing disease. And what we could see, for example, that for high, this cell and gene pairs, which were having a high influencing, were collagen, which is a hallmark of fibrosis, and this was done by MSCs. In TGF-beta secretion, again to megakaryocytes, which is again a hallmark of fibrosis signaling. On the other hand, for example, we saw the 6L12, which is a very important gene for hematopoietic support, so for support of the stroma cells. And this is usually done by the MSCs, not for the stroma cells, but for the hematopoietic stem cells. And this was particularly decreased, and showing also that upon the disease, we have loss of this, let's say, common or homeostasis signaling. So now we could really go from the cell level back to the important players. And of course, what we would like now, also what we explore now, is to look at all the genes here, which maybe are not so obvious, because of course, these are the things which are, for us, the novelty part, right, where we can further explore. Okay, and of course, the other thing we can do is we can zoom in, right? We can really now see, okay, how was the signaling? And for example, if we now look at all the top TGF-beta signaling, and it was mostly all coming from megakaryocytes and going through different receptors, or potentially through almost all the cells in the system. So now we can really do questions to go back in the particular predictions and which were now helped by this kind of topological embedding of the topological measures. Okay, so I think I am starting to go over time, right? So maybe I'll skip this thing, but I just wanted to show, for example, that among other things about, and also by having this expertise in both single cell experiments and cell-cell communication, we apply all these methods, for example, to understand how, because it was known, for example, that patients with COVID have usually, and if they are admitted in ICU, they will have, they are likely to have a kidney injury. And we wanted to investigate this, if this was associated to fibrosis. And we could not only confirm that this, that COVID infect kidney cells, which was already shown before, but we could show this in a more general way using single cell methods. But also we could show the fibrosis was actually the mechanism that was leading to this kidney function loss. And also, for example, looking at the cell communication was also an important way to characterize this. Unfortunately, I have to skip this part, otherwise I would go over the time of the questions, right? And then I just wanted to show that, yes, we can now use this kind of network analysis to try to understand cell communication with the focus on fibrosis, right? And I could show that this was quite helpful, for example, to dissect no mechanism in this bone marrow fibrosis, but actually the same method could also now be applied to kidneys with which were infected by COVID. And right now we actually, together with several collaborators, we're really now exploring several types of single cell or a novel type of sequencing protocols. And here just some examples. For example, we are, for example, now trying to combine single cell and attack and RNA, for example, to try to improve the regulatory networks and to look not only what's happening in the single cell, in the cell communication level outside the cells, but also go inside, because we will know now how potential factors are active and maybe expressing other ligands. Other thing that we are doing is, for example, we have also spatial transcriptomics, so we can now, one problem with a single cell that we lose the spatial information, but with spatial transcriptomics, which are not yet in the single cell level, we can kind of get some knowledge on how the cells are also associated space. And we can do this to, for example, to go to put TF activity, but also do signaling into this spatial space from things that we learn in the single cell level. And for example, particularly hematopoietic diseases, we also try to combine the single cell RNA with single cell DNA. Usually, we cannot do DNA sequencing in a broad way, but usually we can do in target. So we know some variants which are not associated, for example, to NPNs or to myelofibrosis, and we can now try to pinpoint which cells were mutated inside the single cell RNA and see how the mutated cells or these mutated clones might be actually driving the differences in the transcription level. And also, of course, also trying to, for example, derive clone trees. So there is really a lot of things that one can do in the genome level, right, in the techniques, but also this usually requires novel computational methods, and these are the things that we're going to be working in the next years. And a lot of things of many people, right? But in particular, I think this Yanin and Mimbu, who were mostly working in this ataxic part, and James Nagai, which actually is from Brazil, and actually helped to make the contact for this talk, right? Who is mostly working in the cell-cell network analysis. And thank you for your attention.
Okay, Professor Ivan, thank you very much for an excellent talk and for talking to us about these technologies that are really pushing the boundaries of knowledge. We have time for a few questions. A few questions are coming up. First from Aline. What criteria can be used to choose the best clustering method to differentiate cell types?
Yes, I think this is a very important question, right? So I think, clustering was a problem which I have been working on master thesis and my PhD thesis before, when we only have arrays and we were only thinking about, we only have dozens of samples, right, to cluster. And this is a problem, like finding how many clusters is an important problem, and there is no solution, right? So usually what we do is we either have criteria which are kind of heuristics, which kind of help you to indicate this, like silhouette index or stability indices. But in reality, in particular for some of these projects where we are trying to do more the biology, some of this is really a mix of, we really do clustering, and for example, we use now a log clustering, which is a graph-based method where you have a resolution parameter, which you can kind of decrease the number of clusters. And usually the first step after we can take the data, combine in the cluster, is really looking at different resolutions and selecting the one that is kind of reflecting the resolution we want to look at, right? Because on one hand, I show, for example, for the raw data was really easy to find the sub-cell types, right, the major ones, but if you want now to see the ones which are the ones driving the disease, then we need to further cluster them. And this is a process that still we cannot do only in the machine. It is a mix of using some of these clustering determination methods plus interpretation by our specialists, which really look at the markers and say, look, we have these clusters, we can explain. Others maybe we cannot explain, and then sometimes we just merge them by hand because we say, well, there is nothing that we can indicate. Or sometimes we just leave them and say, this is a cluster that we don't know about, and because it's also something that maybe somebody else can explain. I hope I could answer the question, right? But it's a difficult question, which I think there is no clear answer.
Yeah, I think it was a great answer. Okay, now from Maria Luisa. How many patients affected with the disease do you need to generate this kind of data?
Yeah, I mean, so single cell data, of course, is very expensive, right? So when any kind of, so one sample will cost a few $3,000, right? So we don't have many data. So our largest data set that I have right now is with 28 patients. Well, actually not 28 patients, but 28 biopsies of maybe 20 patients. The data show you today, so we do a lot of things based on mouse models, where, for example, in the raw data on the myelofibrosis, we have something like 3 to 8 samples where we have, for example, time courses. We could really characterize changes over time. And then usually we go back to the patient data only for validation, because then we can do only maybe 2 patients, 2 controls. Also, some of these patients is very difficult to get to the, it's not about the price of doing the experiment itself, but also it's difficult to get the tissues, right? And also the amount of tissues that one needs to do the single cell. So, for example, for the myelofibrosis data, we only have actually 2 disease samples. And also for the COVID data in the single cell level, we only had also 1 disease sample. But all the things that we had to validate back, right? So for the COVID, a lot of the things were validated in a larger cohort, because then it's cheaper to do all the protocols where we are only checking on one particular tube, right?
Okay, I think we have time for a last one from Professor José Luis. Ivan, great talk. Did you notice any difference in the expression of endogenous retrovirus-related genes in this kidney fibrosis study?
No, I think it's an excellent question. Of course, I mean, I have been working before on more, let's say, non-coding or non-coding RNAs. Of course, all these single-cell data I was showing are based on the 10x kit, which is only taking polyadenylated RNAs, and we wouldn't know, for example, if the endogenous retrovirus are poly A or not. And actually, we kind of ignored the no coding because we were, let's say, really trying to characterize cells and looking at more signally. But of course, if genes are polyadenylated, one can even go back there and find. And I think I've seen now a few papers really exploring this, and also I think the protocols are improving now. So now we will also soon like to go to protocols where we're doing really full-length RNA sequencing the single cell, and I think this kind of questions can be now answered.
Okay, I think our time is up. Professor Ivan, thank you once again for participating in our event. It was a pleasure.
Yeah, thank you again. Yeah, it was a pleasure.
Okay, bye-bye.
All right.
Bom, pessoal, agora a parte das palestras de manhã foi isso. Nós vamos agora para o nosso horário de almoço e retornamos a 1h30 para as apresentações orais de bioinformática e imunologia. Até logo.